normalmente que van a venir. Sí, a ellos les sale... Y a ellos a veces les salen cosas de... No, no está. ¿Sí? ¿Sí está encendido? Sí, entonces a veces a ellos les salen cosas de último minuto y no sabemos entonces al final, pero creo que en la tarde se llegan más porque la idea era como generar alguna discusión. O sea, la idea de la actividad es un poco como presentarnos un poquito, digamos. Bueno, primero que nada vamos a empezar con una charla del doctor Alex Sheck, que es del MIT. Alex pues es tico de origen, digamos, pero tiene muchos años de estar en el MIT en los Estados Unidos y ya se hizo pues de allá.
Hey, no. Gringo.
No quise decirlo. Entonces, la idea es como hacer presentaciones cortitas o por lo menos de una forma informal, no importa si a mí les dije como que tal vez una presentación de algunas diapositivas, pero igualmente no pasa nada, nada más por lo menos en presentarnos y decir como cuáles son los intereses de investigación que tenemos o qué hacemos de la vida científica, digamos. Pero con Alex sí quería que Alex nos diera como un overview un poquito más detallado de lo que está pasando en el MIT en su grupo, el grupo del doctor James Fox, del cual Alex es parte. Además tenemos a Víctor Jiménez, que es de agroalimentarias, que también está haciendo alguna colaboración acá con Alex en un contexto un poco diferente, pero pues era importante que tal vez Víctor se acercara, primero para que viera más o menos un poco más detallado lo que ellos hacen, y segundo pues porque con las cuestiones administrativas aquí en la U y demás, quizá que es bueno que todos nos apañemos juntos y para lograr cosas más sencillas. Entonces más bien yo le agradezco a Víctor que venga a un ratito por lo menos. Entonces, y ahora la idea es eso, de que vamos a empezar con Alex, después nos va a seguir Daniel nos va a hacer una presentación un tanto corta porque en la tarde y mañana y el viernes tiene varias presentaciones, entonces de ahí se va a ir detallando más un poco en su trabajo. Daniel es PI, Junior PI en un centro que se llama Centro para Terapia Inmune en Cáncer, de un hospital que se llama el Hospital Herlep, que pertenece a la Universidad de Copenhague. Pues vale la pena mencionar, Daniel y yo fuimos compañeros de laboratorio cuando yo estuve en Dinamarca, él después se fue a hacer un postdoc al NIH en los Estados Unidos y ahora pues está iniciando, ya tiene un grupo de investigación, la verdad, grande, ya tiene varios postdocs, ya tiene varios PhD y ya tiene financiamiento importante también, ya ha logrado en 4 años establecer un grupo bastante, que va creciendo bastante. Entonces va a seguir Daniel y después ahí pues un poco nos vamos acomodando, no hice como un orden formal, sino pues un poco tal vez en la forma, yo sé que Vanessa tiene que irse y etcétera, entonces ahí pues vamos haciéndolo de una forma un poco informal, si se quiere. Entonces vamos a empezar con Alex.
Gracias. Bueno, como dijo Warner, mi nombre es Alex, es un placer conocerlos a todos, he podido hablar con... ¿Estamos bien? Bien, ok. Entonces, como dijo Warner, la idea era no ir en ningún tema a gran profundidad, sino darles un poco del sabor de lo que estamos haciendo en el laboratorio. Es un laboratorio muy grande, trabajamos ahorita en muchas áreas diversas y entonces lo que me gustaría a mí es encontrar un tema más que nos podemos profundizar un poquito más, tal vez en conexión con ustedes, porque los temas enmarcados en lo que estamos hablando acá son cosas de mucho interés para nosotros. Pero,
una pregunta,
¿qué
¿será
pasa?
posible que lo hagamos en inglés para involucrar a Daniel?
I can do it in English?
Yeah.
Yeah? Just curveballs. Okay. All right. So basically, okay. So I'm just gonna go over the overview. I'm gonna introduce the work group that Warner mentioned, that I work with Professor James Fox, and then I'll go a little bit about my bio, which is a little atypical for a scientist. Currently, I'm working on a microbiome platform or a service within our group to collaborate with investigators inside MIT and outside MIT. And then I'll just briefly go over a few projects, a recent publication detailing the effects of H. pylori and vitamin K. It's a very weird study, but it was kind of fun. Then a lot of our interests are in these enterohepatic helicobacters. And I'll mention a couple things just because there's a lot of interest in the group about colorectal cancer. This was kind of added at the last minute, but we have some experience in those models. And then some work we do recently with marmosets and just highlights, just touch upon collaborations we've had for many, many years with Colombia and recently more with Nicaragua and Honduras, looking at gastric cancer. And so just to start, this is the boss. This is Dr. James Fox. He directs, and he's a professor at MIT, but he's a director of the Division of Comparative Medicine. Division is normally around 180 people, but it's a very diverse group. We have all the animal care staff. We have the people that do administrative things, the research group, including the postdocs and the research scientists. I'd say is like 20 people, perhaps. But a lot of people do get to work with the research group, so it's not exclusively that research group, but I would say the core for research is about 20 people. The vivariums at MIT are around 18,000 square meters, a daily census about 100,000 animals. There's 15 species of animals, mostly rodents, mostly mice, but we do have the range. We have pigs, we have zebrafish, we have frogs, we have birds, and we have a great veterinary staff that looks after that. And as part of that, DCM has a lot of its own services and cores. We have a histology suite, pathology. We have a transgenic facility to help rederive animals to do microinjections. We have a germ-free facility. We have sentinel programs for surveillance. We do have a clin micro lab. We do aerobic and microaerophilic, which is important for helicobacters and campi culture, and we also have the capability to do anaerobic cultures. And then the most recent addition was the ability to do a lot of microbiome work, doing a lot of sequencing. And so the main interests in the group are directed by Dr. Fox, and it's mostly on the pathogenesis of Campylobacter and Helicobacters, both pylori and then what we call the enterohepatic Helicobacters in both humans and in animals. And I just highlight that, as I mentioned earlier, we've had a lot of success. Dr. Fox is always looking for collaborations, and we've been part of the program project grant that Dr. Correa led till like 10 years ago, that studied a population in Colombia for many, many years. And the other great interest in our group is laboratory animal medicine. What is affecting the study models? Are there subclinical infections? Are there infections that can alter the course of experiments and affect reproducibility in science, or might somehow taint your data? So that's a lot of what we focus on. My bio, I did a PhD in biological engineering. My thesis was on the effect of sex and on the inflammation caused by H. pylori on the mutagenesis and carcinogenesis. And after that, I worked at a startup, a sequencing startup, developing kits to look at hepatitis C virus. At that time, the direct-acting antivirals were coming on board, and they were very specific to NS3, NS4, NS5, but mutations, variants of HCV would not react to certain antivirals. So we were trying to develop kits that would help physicians determine what kind of variant do you have and how it might affect your treatment plan. So I also worked as a scientist at the American Red Cross. We have an HLA lab, which is the human leukocyte antigen. Basically, our lab serviced like all the major hospitals in the New England area, MGH, there's Yale, Boston Children's. We worked with a lot of these hospitals and registries nationally and in Brazil. And what we did is we helped sequence 6 HLA genes to help match patients with donors that are in registries to confirm that they have proper matches and help them figure that out. And my job there was to, for the longest time, it's been done with Sanger sequencing, which is fantastic. Hands down, the product is very good, but the field has been moving to NGS, and so it has some advantages and disadvantages, but I worked in implementing and accrediting that system in a clinical setting. And then I kind of got the bug and I wanted to do some more research. The clinical setting was very kind of repetitive and monotonous, so I talked to the folks back at DCM, and they were interested in setting up these sequencing for microbiome studies. So we came back and we're basically setting up the pipeline to collect samples, advise people on how to do that, and we do the extractions, the library preps, and we send it off to sequence, but then we help analyze the data. All right, and so basically the microbiome platform, the thing we focus on the most, it's pretty easy, is like the 16S rRNA sequencing, and that basically answers the question of who is there. And just, it has started in the last couple of years, and we're trying to get out from just who is there, which is just gives you a big list of things that you really don't know what to do with, but use the tools in the division. We have a lot of work with targeted qPCR. We do a lot of fish. We have experience in cultivation of aerobic and aerobic and all sorts of bugs, and try to see if we can push out and take whatever we understand from the microbiome and actually get functional knowledge on diseases. And so that's what we're trying to build. We have a little bit less experience with metabolomics or proteomics. We have done some transcriptomics and metagenomics, and here I didn't put it in this slide, but we do have the germ-free facilities, which gives us an advantage. We've mostly used the germ-free facilities to mono-associate bacteria, but a lot of people in other places use consortia of bacteria to see how those affect things. And because we have the transgenic, we can rederive all our lines, have them be germ-free, have them raised in our germ-free facility, so we can have whatever model we have raised germ-free and then associated with specific bacteria, which we have done with the NSCAS mouse line, which I will discuss further. And so, yeah, that's kind of what we're talking about. What is the next step? Where can we take this very basic first observation of what the microbiota is, and then how do we take that further? And that's kind of where we always, every experiment is obviously different, and we're trying to see how we can figure out how to do that in the context of our collaboration here. So I'll just jump in, touch on a couple studies that are more recent, just to give you a flavor of how, in a sense, we're diverse, which is good and bad. So this is the NSCAS FBV mouse model. It overexpresses gastrin. It naturally will develop a gastric cancer-like phenotype, dysplasia, in 12 months, but we can accelerate that progression by incorporating Helicobacter pylori. So we'd like to see what is it about Helicobacter pylori and the inflammatory cascade that accelerates that. We've worked in this model for 15 years, 20 years. So there was a veterinarian that wanted to set up a new study. She just kind of picked two papers that we'd done. One was eradication of H. pylori with antibiotics, and we had shown a long time ago that you can reduce the high-grade gastrointestinal neoplasia as scored by pathology. And then there was another study recently that was more of a dietary study where you increase folate levels, and the hypothesis there, and what we showed in that study is that if you increase the dietary folate levels early on, you have a more protective effect, you have more DNA stabilization, and it has to do with methylation. So she wanted to see if some kind of late antibiotic intervention combined with an increased folate diet would be synergistic in some help. So this is the study we set up. It was pretty basic, and she was hoping it would go well. Unfortunately, there was like weird unexpected effects due to the synergy of the two treatments. This is a mouse. As you can appreciate, it's very not normal. The mouse is, like, if you looked at these mice without opening them up, they were extremely pale. You could actually see a blue tint, and so that was a bad indication. They were lethargic. When you open them up, this is the liver, which is very, very pale for the liver. The texture of the liver was wrong. And this is the GI. And the GI is full of blood. This is zoomed in. There's the liver again, and this is the stomach. And basically what the veterinarian was able to assess clinically is that it was full of unclotted blood, which she tried to demonstrate in that smear there. And so these mice were having unexpected mortality, mostly in the group treated with antibiotics. So 42.8% of the mice did not make it to the end of the study. 37% of the mice. And this is a model we've worked in for decades. You know, we normally run these experiments for 8 months, no complications. You lose a mouse there, here and there from a study of 100 mice. But this one we're seeing extremely high mortality associated highly with the antibiotic treatment. When you do like clinical panels, the hematocrit is way lower in animals that visibly were affected, that had that blue tint, compared to cage mates that were relatively healthy in terms of the blood parameters. So at this point, being veterinarians, they figured out that it was probably it was a hemorrhage. That was normally associated with vitamin K deficiency, so there was an intervention using phylloquinone or vitamin K1. So we took measurements pre-PK and at the end of study, and you can see that all these groups that were affected were able to be restored. And so this is kind of where it was interesting to us to dive deeper, so we started looking at what the synergy was. So normally the diet will include a normal conventional mouse diet will have lots of plant and unknown materials. It's not a fixed diet, but this was a semi-synthetic diet, so everything was chemically produced. And so there was, when we talked to the nutritionists, they said that there's like no extra vitamin K, so the level of vitamin K that is present in the diet was very marginal, I guess was the correct word they used. So normally in a mouse, there's two forms of, or in humans as well, there's two sources of vitamin K. One is in your diet. If you eat your vegetables, you'll be fine. You won't have to be scared about this. And normally the bacteria in your gut, because they use menaquinones as part of their electron transfer chains, they actually synthesize vitamin K2, and the body can absorb that and use it in times of need. And in humans, people that have been on a lot of antibiotics, you can see a reduction in vitamin K. But normally with people, it's easy enough to eat something and you're fine, right? And so what we did is by changing first the diet, making it semi-synthetic for the folate part of the study, and then applying the antibiotics, we're able to knock out both legs from under the mouse. And both previous studies had not seen any of these effects, but it was this combination of things that kind of affected the mouse. And so we wanted to pursue that further and kind of see exactly what changed in the composition of vitamin K. And what was surprising to us is that the total vitamin K was actually relatively stable. Like if you combine, this is looking at the feces, and our colleagues were able to analyze the feces, and these are all the menaquinones, vitamin K2s created by bacteria. And you can see that, you can't see it in this graph, but like when we sum all the vitamin K, it was pretty much the same. But what we noticed is that if you have a regular chow animal, they don't have the phenotype, and these two animals, they look slightly different, but none of these three have the phenotype. The major difference that we saw in the antibiotic-treated mice was this reduction in MK6 and MK5. So that kind of helped guide us in a way to specific bacteria that in the literature are associated with the creation of MK5 and 6. Specific bacteria can only create certain menaquinones based on the side chains they add at the end of the process. But that also in itself is not very well characterized. When we looked at the microbiome data, we saw a drastic decrease of Bacteroidetes, and this is at the phylum level, an increase in Firmicutes, and some increase in Proteobacteria, and the Verrucomicrobia was very noisy. We did observe changes in diversity. Obviously, when you give antibiotics, you wipe out so much of the diversity. But the most interesting part for us, and the reason I want to mention this is just trying to show this as an example of how you can marry different types of data that are not commonly used. And this helped us narrow it down to the pathways. And so using in silico analysis called PyCrust, we're able to predict what are the bacteria that have specific pathways associated with menaquinone synthesis. And then from that prediction, we were able to pull out which are the bacteria that were highly reduced that have the menaquinone synthesis pathway. And we came up with these three candidates. We were able to develop qPCR to look directly at the genomic copies of this gene called MEN-A that is the last step in the menaquinone synthesis pathway. And we were able to show that in a lot of our antibiotic-treated mice, and I told you these varicomicrobia were very bipolar in a way, but you can see that a lot of these groups got drastic reductions, and this is kind of where the study ended up right now, but it gave us future avenues because we are very interested in figuring out, if we proceed with the study, like what are the organisms that are very specific to the production of MK6 that were affected in the study, and how the loss of specific consortia would affect this phenotype. I'm going to switch gears very quickly. I apologize. So this is studies done in, I believe, NIH by Barbara Rehrman's group, by Ross Hart et al. And they had been interested for a while, based on some findings out of Minnesota, at looking at more feral mice populations. And they have talked a lot about how the immune system of a more exposed mouse will have a more reproducible, is more similar to what humans have. And laboratory mice are very much like what a neonate would have in terms of their immune component. So this is basically showing a wild mouse caught near Maryland. These are wild mice that they transferred the microbiota, and they can maintain this microbiota, but these are lab mice that were bought. And you can see that there's drastic differences between this section and this section. When you look at it plotted on a heat map, these are your wild or wild-related mice, and these are your lab mice. And this always catches our attention, is that Helicobacters are the organisms that we preach should be always excluded from a laboratory setting because they have ill effects on your research, and indeed they do, but now there's a lot of interest in actually incorporating the diversity of Helicobacters and how they might affect your research animals. And so you can see that Helicobacters are highly abundant in these wild mice. So this is actually a very old study that we dug up, that we wanted to look up because we have a lot of archival tissues and frozen tissues. This is a very interesting study where we had a very basic H. pylori model with the black 6 mice. You can induce a very robust inflammation, no cancer. But if you underlay a single bacterium before you infect the mice, so in this case we use two. We use Helicobacter muridarum and Helicobacter hepaticus. So this is always the observation. When you put a Helicobacter in a mouse, it's a completely subclinical infection. There's no effect. You pick those mice up, you can't see them, you open them up, there's nothing different. There's no pathology difference. But when you put the Helicobacter pylori infection on top of that, the muridarum somehow diverges from the hepaticus. So hepaticus has less effect. We use hepaticus a lot because it's more characterized in the literature. It's considered a pathobiont. But muridarum somehow is very protective. In other papers, they've shown that it can alter the secretory IgA pool, and it can do selective pressure on the microbiota and change what you observe there. And so a lot of the cytokine expression is also different. This one is interleukin-17 is reduced in the HMHP group. And so this is a study that was done in 2011. We didn't have the capabilities we have now. So going back to the archival tissue and the frozen tissue, we've tried to sequence, and this is the stomach from these organisms. So these are mixed, but this is Hepaticus muridarum at 7 months, and the first quarter would be H. pylori infected as well. And this is Hepaticus and muridarum, and you can see that starting from the same colony of mice, just giving them a single organism, you can completely cause a divergence in the microbiota in the stomach, and that will have repercussions when you overlay the H. pylori infection. So this is an active study that we're trying to follow up and look more into, but the stomach in the mouse is mostly either some sort of lactobacillus, but it alters which flavor of lactobacillus you get. So Hepaticus induces a much higher abundance of Reuteri, which some people propose as a probiotic, but for some reason these are not as protective as the ones that do not have Reuteri. And just to keep going with this EHS story, these are older studies, but I think it might be interest for the group because we talked about colorectal cancer. So this is a model we've used for many, many years. It was part of the big nitric oxide grant that MIT had with like 5 PIs. This was the workhorse model that they used for many years. We were able to show that through cytolethal distending toxin incorporated in H. hepaticus, which I tell you will mostly cause subclinical effects on the mouse, but when you look very closely, you can see that there's an induction of DNA damage. So and that's mediated through INOS and IL-22. So if you add H. hepaticus to these, so these are RAG2 mice, they lack B and T cells. So you can induce a lot of INOS, you induce a lot of proliferation seen here by Ki67. And you can also, recently we've developed this gamma H2AX that measures double-stranded breaks. And you can see that this is all correlated with, associated with the H. hepaticus infection. But when you treat with NMA, which is an INOS inhibitor, when you treat with anti-IL-22 antibody, you can reduce a lot of the INOS, you see less proliferation, and you don't see as much change in gamma H2AX or double-stranded breaks. This model is characterized by an accumulation of neutrophils and macrophages. And since it's a RAG2 deficient model or RAG2 knockout model, we also have done adoptive transfer studies way in the, a couple of years ago. And they found that if you transfer Tregs into this model, you can inhibit the formation of the tumors that we see. It's very interesting. We've done different crosses with APC men, and in that mouse, you have tumors in both the intestine and the mammary glands. I have not covered all this data. Oh, this is a recent study also. We use a technique to measure mutant frequency. So you can actually excise specific pieces of DNA and sequence those using Sanger sequencing. And that could actually tell you the profile of the mutations. I think now that technique is getting a little bit older because just with a lot of sequencing being a lot cheaper, you can sequence much more deeply. You can do and get mutational signatures, which is what they do for a lot of patients now. And these are two different helicoenterohepatic, or actually these are from the same paper. So this is the H. japonicum, but we've shown another Helicobacter seguini from tamarins. And if you put them in IL-10 mice, because the RAG2 was a very kind of brute force model, and now we've focused more on knockouts with just interleukins, we can cause dysplasia just by adding a single organism in these models. And so these are things that we are interested in, looking more at how these organisms can affect the microbiota, how they affect the immune environment. And this just got thrown in there. This is PKS E. coli. It's another interest of the lab. I'll probably talk about it more on Friday, but PKS E. coli has been very hot recently. We've been doing a lot of surveys in our facilities for it because it's associated with incidence of colorectal cancer. And recently in 2019, there's been 2-3 very high profile papers where they've actually been able to isolate colibactin and address how it does the damage, how it figure out the structure and see how the mutational signatures of colibactin and show that it is indeed causing damage directly based on the presence of this E. coli, and that it is an important factor. And I believe, oh, nope, sorry, there's 2 more sections, I apologize. So we're gonna talk a little bit, change gears again. We have a lot of interest in neuroscience to use different models and that you can genetically modify. This is the Calatrix jacus. The translation says it's titi común, but in Costa Rica we have monos titis, which is not the same. So I call them marmosetas. So these are Brazilian monkeys. They are about the size of a rat. In the wild, they eat gums and insects and fruits. In the wild, 30 to 70% of their foraging activity is getting this gum. And so this is the work I've been chatting with Victor about, just learning more about plant products and how they might affect diets. But they, in our hands, and then actually in labs worldwide, all of them, 10 to 20% of them develop IBD basically, a chronic wasting syndrome. The marmosets will continue to eat, but they lose weight, they have low serum albumin, they have a lot of body condition problems, and ultimately you can treat them with glucocorticoids, but that's like a lifelong treatment, which makes it expensive and affects research. So there's a lot, our lab is very interested in helping ameliorate this problem. They have chronic diarrhea and colitis. So this is work, and so basically we're trying to see what the correlations are with IBD. IBD is a very important subject. Recently, a lot of work has been done at the Broad and the MGH. It's 1 to 3 million individuals in the US, but it's rising worldwide. People believe it's associated with industrialization or westernizing of diets. Roughly it gets broken down into 2 very big bins. One is Crohn's disease, one is ulcerative colitis, but we really think there's a much bigger spectrum because basically if it's in this region, it's UC. If it's all over, it's CD, but it could be lots of different diseases that we have just not typed correctly. And basically, this chart is kind of more of a joke to me. Like, basically, everything gives you IBD. So nobody knows, and there's a lot that we're working on. There is some indication that it is related to the bacteria because there is an initial response to antibiotics, but that is lost because then the bacteria either gain resistance or you select for the wrong populations. You can, in CD, do some surgical intervention to deviate fecal matter, and that alleviates some of it. And then if you take these organisms and put them in germ-free animals, you don't see as much of the effect. So, or that didn't make sense at all. The models that are germ-free do not show the same potency as the models that are conventional laced. And so again, this is just a soup, big pile of things that have been associated with IBD, which is a lot, but a lot of it is immune mediated. They think a lot about autophagy and how responses to bacteria are being co-opted and become more inflammatory, and they lead to this damage. In terms of the microbiome, this is just a quick summary of how people have approached the problem. I mean, we talked a little bit about vitamin K, but the microbiome metabolizes a lot of different nutrients that might be protective. A big focus is on short-chain fatty acids. These are polysaccharides digested by the microbiome. They can directly interact with our host cells and stimulate different types of immune compartments. And the microbiome also generally protects against pathogens. And this is just a review paper that looked at different papers linking IBD and the microbiome, and you can see the list is pretty long. And again, this could be that problem that we're using a very big blanket term to describe many different syndromes. But in the marmosets, this is what caught our attention, that if you look at the W's here, these are wild marmosets, and these are not all Jacus, but there are hybrids or different species. But you see a lot of actinobacteria or bifidobacteria, basically. That is very commonly associated as probiotic. But if you see in this study, the more captive marmosets start having more gamma proteobacteria. Epsilon proteobacteria is all Helicobacter, pretty much. And if you look at 3 different institutions, 2 in Germany, 1 in the US, you don't see actinobacteria present as high as you see in the wild. And we think it's because when you translocate it, and this is something that has been seen in lots of other captive species, you don't recreate the diet, and marmosets actually are worse off because they're obligate gum feeders. So they really spend a lot of their time, they have adaptations in their teeth to actually find this gum and get it out of the trees. They have adaptations in their cecum that makes it larger so that they can be gut fermenters. And when we just give them dry chow and lots of fruit, you're giving them the wrong sugars, you're giving them the wrong conditions, and it might be associated. And it has been hypothesized. Nobody has had the time to really test this hypothesis, so this is something we're starting up this year. These are our colonies. We have actually lots of animals sampled over a long time. We have 5 different sources represented here. So these are different institutions we've either acquired animals from, and you can see that each institution has its own unique microbiota. And this is the institution where all our animals have the most IBD. You can see their microbiota is very different, so we're trying to figure out how do we take that data, how do we correlate it to our clinical parameters, how do we narrow down the potential causes, and so So that's kind of where this is going. This is just a very simple PCA analysis. And what we're looking at is like all our healthy folks, mostly clustered here, are the folks that respond better. And like if you look at just their bar graphs, and this is like a very not cool way of looking at this. This is not sophisticated at all, but if you just eyeball it, you can see big differences between sick and healthy animals. We have been toying around with things, but like our random forest analysis can very well predict which source it is based on using a lot of these features, but we haven't been very successful in classifying the disease progression, and that could be in part because it's really hard to classify. I can go to a clinician and they're like, well, this animal has diarrhea, but it's really, we don't have a gold standard clinical metric. A lot of people use weight and albumin, and so that's a problem right now. So we can't really bin things cleanly for this kind of analysis yet. And lastly, I'm just going to touch on just work we've done. We've published lots of papers and worked. We're very happy with work we've had with Vanderbilt and LSU in the past, looking at 2 populations of gastric cancer patients in Colombia. We looked at people in the mountains and people on the coast, and just seeing the genetic components and environmental components. One of the interesting findings was that people on the coast had a lot more helminths or parasites that might affect their immune response to Helicobacter pylori, and that was a while ago, and this project is starting to has wound down a lot, and now we've moved to looking at populations in Nicaragua and Honduras, where León is our low gastric cancer incidence region, and there's a population in Honduras where they have a high incidence of gastric cancer. And just putting it out there in terms of, so we have done some of the microbiome analysis. This is kind of more of a cautionary tale than data. These are samples we received from Nicaragua, and you can see that it's all over the place, but this is actually what we found later is laboratory error. We had lots of supply or freeze chain problems on the way up. These samples apparently thawed, so this is just something like, while we're talking about these contexts, we have to think a lot about the logistics. And they had a freezer go down, they had problems with electricity, or problems in shipping, or it went to Vanderbilt first and then to us, and so there's a lot of exchange of hands, and like you can see that the sample changes a little bit and it completely throws off all the analysis. Staph is so resistant and easy to culture that it just overbloomed and completely destroyed the integrity of these cultures, and most of these cultures we can't use, unfortunately. So it's just throwing it out there as a cautionary tale. This project is still ongoing. It's kind of going slowly, but it's something we're very interested in pursuing. One couple tidbits, like we didn't know what to expect, but when you look at individuals that are samples that we can trust, like the self microbiome is much closer to itself than the same site in several individuals, and that has been shown in the literature recently. But so if you look at the antrums across the population, we didn't know if there would be an antrum-specific microbiome versus a corpus-specific microbiome, but it seems like your self will override the site in this case. And the other thing we kind of looked at briefly is just a simple regression, looking at the phyla of different people stratified by their clinical diagnosis. And if you look at everybody, you don't get like a great R-squared. But when you look at the people with just gastritis, it lines up much nicer. But the people with intestinal metaplasia, you start seeing a divergence. So we're wondering if that is part of the pathological process where the microbiome is indeed changing, that maybe some parts of the stomach are starting to not be as favorable a niche for the normal microbiota, and that it's causing some change. And just to conclude, these are my acknowledgements. This is for the group at MIT. In one of the projects we worked with the folks at Tufts. We work a lot with the group at Columbia and Vanderbilt and UAB now with Doug Morgan. And just very excited to be here. Thank you for your time. And we love to collaborate. We love to be a nexus to people. I'm very excited to see all the stuff you guys are doing, and mostly are interested in how we can help in microbiology, GI disease, cancer, And using whatever facilities we have, we're happy to talk about how we could use them to help your projects in animal modeling, germ-free, in the microbiome, bacteriology, etc. Cool. All right. All right. That was way longer than it should have been. Sorry. Oh, yeah. That's the one I do the least, Daniel, but I am happy to. I have.
No, there's just some things that I think it's maybe a stupid question, the things I didn't really understand. So you use the ractobacillus because that otherwise they clear the helicobacter?
Yeah, I think. I don't know if they clear it, but you don't develop anything in a conventional mouse, a wild type mouse. Like with Helicobacter, they're subclinical. They're perfectly healthy. They'll live forever. They don't clear H. hepaticus, though.
Okay.
Yeah.
And I didn't really get, so the cancers you're getting is
Oh, that didn't work.
it doesn't matter. Is it colon you're getting them or?
Yeah, I think they are colon cancers. Large intestine. Yes.
And how do you administer the bacteria? Is that just all?
Oh, yeah, it's just oral inoculum. We gavage it 3 times. We grow it up, and they can, yeah. There's nothing complicated with the bacteria. It's not injected. It's just eventually it will make its way down. It lives in the lower bowel past the stomach, but I think mostly in the cecum.
Okay.
Yeah.
How do you screen if they're colonized properly by PCR on feces? Or?
EHSs are easier because they live further down, so you can do PCR. Yeah. So that's kind of like the bread and butter of the lab, is just looking for animals that have helicobacters, and we were actually very successful in getting all the vendors to eliminate helicobacters, and now everybody wants them back in. And we're like, oops, that was our fault. Yeah. So.
Regarding the studies that you are performing in Nicaragua, you mentioned, well, of course, a lot of logistics and problems regarding to the freezing and things like that. But what you were measuring, or the microbiome analysis that you are doing, is it on fecal samples?
No, it's directly in the stomach. They're biopsies, corpus and antrum. But the limitation there, not limitation, but like why this project here is more interesting is that over there, most of them were just gastritis. The worst pathology we saw was intestinal metaplasia. That could be because it's the low risk area in Nicaragua, but it also be that it's just the patients they're screening. It's not like people with gastric cancer. It's just people that come in for a screen, and those are the samples we get.
And do you get biopsies from different parts of the stomach?
Yes, they take like 7, but like it's kind of that thing where everybody gets a piece, so we get very small portions of each one. So they have to culture them in Vanderbilt. They do other processes. So, and I think they also want to keep some for molecular analyses. So we get a small piece that we can, basically what we do, we macerate it, we put it into culture media so that we can do anaerobic and aerobic cultures, and then we also do the microbiome from what is left. So we extract DNA from what's left. Yeah.
What would you expect if we get, for instance, now in the project that hopefully we will start soon with biopsies from gastric cancer patients? We are supposed to collect from neoplastic and non-neoplastic tissue.
Yeah.
Would it be kind of less interesting, or what?
Well, I mean,
Expectation we can get out of it?
the literature in gastric cancer has been, there's like half of them show lower diversity in gastric cancer than other regions. In colorectal cancer, you do get very different microbiota sometimes in adjacent tissue than, so I think I would like to see if that's the case in gastric cancer, basically. And so yeah. Yes, Bob.
I think it's very interesting you mentioned, if I understood it correctly, that your individual profile of the microbiome is like stronger than
Yes.
the site profile.
Well, at least in the stomach. So, yeah.
But that's very interesting because then you can go to populations and see too if certain populations have different microbiota than others. And
Yeah.
draw conclusions from risk of cancer or other diseases.
Yes. And that's part of the hope, but But that conclusion is something we would like to see more. We found it in the literature mentioned that, but it's also like the stomach is a big sac, right? So is it just constantly somehow mixing, or could it be iatrogenic? Like in the person doing the biopsy, what was the protocol for sterilizing the device between, because you're punching it continuously. So that's something we should look further, but it has been supported in literature and it does seem to show up here.
I have followed all the studies that Dr. Fox has performed with these mice that have non-hetero or kind of other non-pylori helicobacters, and then when you mix them, I think you mentioned that the Helicobacter pylori plus Helicobacter hepaticus, then you see increase in cytokines, for instance, interleukin 17 and things like that. Is that recapitulated with some kind of non-hepaticus helicobacters? Do you expect to see different figures, or have you done anything?
We have discussed doing, like we have a very big collection of helicobacters and trying to see which ones do it. We did do it once with Bilis. And I'm blanking on the result. The Bilis experiment was actually earlier, but when we went back to the tissues, that's why I didn't show the data. I think they were older and I think maybe more degraded because the data was very awful from that group. Yeah.
Because as part of the things that we are going to perform or to do together, it's a project in which we are, because we found here that our animal facility is kind of generally contaminated with 4 different species of Helicobacter. That's why when we brought the C57 black 6 mice, we had to isolate them completely. And we actually, with your help, starting to monitor by PCR analysis that our black 6 mice were not contaminated as compared to the other colonies that are completely contaminated. And one of the projects that we are going to do is to characterize the microbiota in mice that we intentionally contaminated. We put some black 6 mice to live on cages where there were mice that were living there that were contaminated. So they were intentionally contaminated, and we compared them with mice, black 6 mice that were only having Helicobacter pylori in the stomach. So I guess it would be interesting to see.
And that's something we're currently trying to see of how early on like these changes might occur, and how, because there is a very interesting paper out of Penn where they just H. miradarum alone was able to help Jackson mice perform similar to their normal house flora. And that was the one that I mentioned. They use IgA and then just use IgA to tag all the bacteria that is in the feces. You pull out everything that is IgA tagged, and you can kind of see what the mouse is looking for. That was a pretty neat technique that is being used now to kind of see what the mouse is selecting against. So that's pretty, but it is obvious that miradarum alone could have that effect on changing the microbiome.
There are no more questions?
I apologize, I need a clock next time.
No problem. I mean, I think we have time. Francisco mentioned that the coffee was about 10 o'clock or something like that. Just be sure that it's ready. Otherwise, we can actually, because Alex and Danny have not been around the PRIS-Lab, so perhaps we can make a small tour while the coffee is ready, and then we take the coffee and then we keep going with Danny.
Do you hear me?
Yeah, awesome. Thank you.
Good. All right, I'm not going to talk for a long time. I'm just going to give a very brief introduction to the research center where I'm working, and just a little bit about what I'm focusing on and what sort of the main techniques and the methodologies we're using in my research group. So just to introduce myself first, I'm a junior group leader at this research center. So I have my own small research group there, and I'm actually also head of the animal facility at our hospital. And luckily, I didn't bring any numbers because they wouldn't really compare to Alex's numbers at all. It's a very small animal facility. Number of species in our facility is 1. We just have mice. So I'm not going to brag about that, but it's something we have, and we use it a lot for the research that I'm doing. The center I'm located in is called the National Center for Cancer Immune Therapy. It's in Denmark, just outside of the capital Copenhagen, at one of the biggest hospitals in Denmark called Herlev Hospital. And it's a complicated structure with actually 3 directors, but it doesn't really matter that much. They each have their own research group. In addition to that, we're 2 junior group leaders. So the whole research center is comprised of 5 research groups. And we also have sort of a core facility at the hospital for doing flow cytometry and also for doing different types of cellular therapies, especially a type of immunotherapy that's called adoptive T-cell transfer or adoptive cell transfer. I'll just mention that briefly later on. Just a little bit more about the research center. Okay, not really responding here. Sure. So initially the research center was very much about clinical trials, and then later on it's been broadened out a lot to really also cover a lot of basic research. And we're really trying to sort of bridge the basic and the clinical research in many different ways. So we have lots of clinical trials going on at the research center. A lot of it is related to this type of immunotherapy, adoptive T-cell transfer. I don't think I have a slide here, so maybe I can just mention it briefly. It's a type of immunotherapy where the principle is that you isolate T cells from an excised tumor or tumor biopsy, and you grow the T cells, you expand them to hundreds of billions of T cells that you reinfuse back into the circulation of the patient. And it actually works very well. The response rates are comparable to anti-PD-1, which is one of the most successful of these checkpoint inhibitors. But the downside to the treatment is, of course, that it's requires a lot of work power and manpower to do this, and there's a limit to how many patients we can treat each year. But the principle is working. So, as I mentioned, we're covering a lot of different topics, all the way from basic to translational to clinical research, and it's kind of listed here. But I think the take-home message is really that we really cover all aspects of immunotherapy research. It's vaccines, it's checkpoint inhibitors, combination with other treatments, it's basic research to identify new immune suppressive mechanisms that can be targeted. My own focus is on the tumor microenvironment, how that affects immune therapy. Lots of different types of research questions. And what we try to promote at the center is really this ability to have sort of this loop where you have the clinical research, the trials. We collect a lot of samples from these trials that we then use to analyze and to generate new hypotheses that we can go back into the lab and test in vitro or in mouse studies, and then hopefully based on that start new clinical trials that are based on these new results to maybe improve the treatments or come up with completely new types of treatments. So my own research, as I mentioned, is about the tumor microenvironment, and that's what we call everything in the tumor that's not the cancer cells themselves. So it's all the healthy cells, the stromal cells, the immune cells present there. We especially have a focus on fibroblasts and macrophages in the tumors. They're also some of the most abundant non-cancer cells in tumors, or in solid tumors at least. But we also have a big interest in extracellular matrix proteins, or connective tissue proteins, such as collagen, which is often present at high levels in tumors, and also other non-collagen extracellular matrix proteins, how that can affect T cells in the tumors and how that can affect the efficacy of these immune therapy approaches. So we, of course, I'm not going to go through all of the different projects we are working on right now, but we have quite a lot of projects going on in the group with trying to address how specific components of the tumor microenvironment affect T cell activity. Especially, we've been focusing a lot on collagen in the tumor. We have studied ex vivo and now also in mouse studies if that can have a direct effect on T cell activity and on T cells' ability to kill cancer cells. But we're also studying how it affects other cells in the tumor microenvironment, how it affects macrophages, which we already know can have a huge effect on suppressing T cells in the tumor microenvironment, if there's sort of an interplay here. We're trying to see if other ECM proteins actually do have sort of an immune suppressive function, more sort of a screening approach. And then we have different projects where we are trying to deplete certain subsets of either macrophages or fibroblasts in mouse models, and in this way see if that changes the response to checkpoint inhibitors. And we're also moving into different PDX models, so trying to use some of the clinical patient material and graft mice with this cancer tissue to grow tumors in mice, and try to model some of the treatments we're doing with the patients as well. So as I already mentioned, we're in my research group, we're very much using different mouse models of cancer, both very simple syngeneic models where you have a cancer cell line, you grow it in culture and you inject it subcutaneously typically. We also have different transgenic models where we genetically induced cancers. And then we have different other types of transgenic mice, some where we can deplete different types of cells quite specifically in the tumor. And more than that, we of course also use a lot of the primary cells we can get our hands on. We can easily get them from mice, but also sometimes from patients, and do ex vivo studies with these. And we try to do this often in 3D cell culture systems to at least avoid some of the cell culture artifacts you get on a plastic surface. And the techniques are quite standard techniques for sort of an immunology lab. It's a lot of flow cytometry, it's a lot of immunohistochemistry, and we have good collaborators that help us do a lot of RNA sequencing. That's an important thing for us as well. And then we have a lot of sort of more classical immunological assays where we can test T cell activity, either directly looking at their ability to kill cancer cells or looking at other sort of pseudo markers of T cell activity. And I think that was it. This is my research group at the moment, except that Dennis, he's a master's student, he left, so now, oh, is there more? Yeah. So now we have Claire instead. So it's a couple of postdocs and a couple of PhD students and some master's students. And I actually think that's what I brought here. But of course, if there are any questions, you could just ask. Sure.
It's more of a logistical question. The National Center for Cancer Immunotherapy, is it under any university or how does it work?
If it's at the university?
Yeah, like, are you associated with the university or are you government run?
Well, so the hospital where we're located is part of the university hospitals in Copenhagen. But, well, it's a bit complicated because actually there is quite a gap between the university and the hospitals. So we're not directly a university center, but it is a research center, very similar to what we would find at the universities. It's just located at the hospital. And it was just called the Center for Cancer Immunotherapy, and recently it became the National Center because we got a large grant. So we sort of have an obligation to also collect information from all the other immunotherapy that's going on in the rest of Denmark and from the clinical trials in other parts of Denmark. And also that a lot of it is focused on the clinical trials and in sort of educating the medical doctors that are using these new types of treatments, because they are encountering new types of adverse effects that they need to know how to handle. So we're kind of trying to use each other's experiences, because it is a bit of a new treatment with completely different side effects. Yeah.
That's a very naive question. So in terms of, like, if you have two patients with, you characterize the exome and you know kind of a mutation that is driving the cancer, with the tumor microenvironment, if you put it, if you use it as a PDX, would that be recapitulated, or is it deterministic or stochastic?
Well, no, you're not going to recapitulate the tumor microenvironment, except if you take and you don't grow the cancer cells or isolate them, but you take the entire tumor tissue and implant that. Then it's been shown that at least for 1 or 2 generations, you will keep the original tumor microenvironment.
And how about between patients? Like if it's similar genetic mutations, would they present in that patient similarly?
There is some correlation, but it's not very strong. So I don't think it's purely the genetic profile of the cancer cells that drives how the tumor microenvironment looks.
The host.
Yeah. So, and then there's of course what we're very interested in is this whole thing about that often the cancer cells, they will start to express some new antigens, either mutated antigens or overexpress things that are not supposed to be there, and that makes them visible to the immune cells. But that is something that also varies a lot between patients, and I think it's also often quite random if the T cells, for some reason, they start to recognize them as foreign or not. So I think the immune system, I think, plays a huge role in how the tumor microenvironment is going to end up looking. Right? Yeah. Yeah, sure.
In the RNA-seq expression analysis is in the immune cells or in the cells related with the matrix extracellular?
So a lot of the RNA-seq we're doing is on total tumors. So that will be cancer cells and the stroma and all the immune cells there. So it's sort of a bulk RNA-seq. And we often actually use it as sort of a way to predict which types of cells we have present in the tumor. Sometimes we also do FACS analysis of certain cell populations. We have the possibility of doing single cell sequencing. It's just so expensive. So often we end up sorting out cells instead and sending them individually to RNA sequencing if we're interested in certain cell types. And we've mostly been doing that with macrophages so far. But we would like to do it for all the different cells. Yeah.
Let's give a chance to Jose, because Jose has to leave for a while and we'll come back later on. So now we can actually show what he has been doing with bioinformatics.
Thank you. Ready? It's ready? Yeah? Working? Okay. Thank you so much. I'm Jose Molina. I'm researcher at Centro de Investigación de Enfermedades Tropicales. I'm a bioinformatician, but to make this possible, I finished a degree in mathematics, another one in microbiology, clinical chemistry, and now I'm finishing the PhD program in science. Basically, My experience in the wet lab is focused on different models in both prokaryotic and eukaryotic models, but working in different techniques related to molecular enzyme biology, microscopy, NGS. But actually, I'm really focused on dry lab, basically working in different pipelines related to data analysis, including bioinformatics, systems biology, and machine learning. In different levels, including genomic and transcriptomic level, and then integrating phenotypes in order to create multi-omics models. Basically, more important for prokaryotics models, we have different pipelines related to de novo genome assembly, but they can also use for metagenomics. In the structural analysis, we have pipelines related to copy number variation or variant calling, pangenome analysis, or the identification of new antigens that can be used to map or to track differences between tumors, for example, comparing with normal tissues. In the case of transcriptomic, we have different pipelines related to data analysis of microarray or RNA-seq, including differential expression or co-expression analysis. And basically, we can put all this information together to build different networks related to, specifically in the topic of systems biology, in both bottom-up or top-down systems biology approaches. For example, in the case of bottom-up approaches, building small world networks, for example, for a specific pathway, and then try to correlate with different phenotypes. And in the case of top-down systems biology, basically using this data, microarray or RNA-seq, we can build different large-scale networks, identify hotspots or hub genes in that networks, and then try to modulate the response in that cases with specific genes. And we can also integrate metadata using different machine learning algorithms in order to finish different pipelines related to clustering, classification, gene selection. So I'm going to show some examples, some of them related to prokaryotic models, but the idea is that we can implement similar approaches for other studies, including cancer. For example, this one is related with the de novo genome assembly with a prokaryotic model, but we can use that information to create pangenome analysis. So we can also use this kind of information to compare different metagenomic data. So I don't really have the experience in a specific project, but I know that we can use the same pipelines in order to have or to compare different profiles. That's basically that with different pipelines related to metagenomics. In the case of variant calling benchmarking, we have implemented different variant calling pipelines related to identification of SNPs, for example, in DELs, in different genes, and we can use this for the identification of new antigens, and we are working in different pipelines related to in a first moment, we are working with public data, but the idea is then in the future try to implement with our own data. In the case of transcriptomic analysis, we can implement classical RNA-seq analysis, including identification of differential expressed genes, and then try to build different networks, large-scale networks, and then identify the hotspots of that network, as I was saying. This is basically the same in this case using different time points along time. And we are doing similar analysis with specific works related to cancer with Adrián Cabezas y Lucía Figueroa in coordination with Professor Warner Alpizar. We finished another project related to screening of RNA interference with cancer cell lines, basically in a similar approach to identify differential expressed genes to build the network, and finally to identify the hotspots. And now the team is working on the modulation of the genes to eventually correlate with the phenotypes. And finally, I guess we are finishing. We have different approaches related to machine learning to identify differences between different biological conditions. In this case, using expression data to identify or to rank the genes by importance, and then to map these genes using different networks. And finally, this is an example of a bottom-up systems biology approach with Dr. Rodrigo Mora. Here we're working with the sphingolipid pathway to correlate this model based on differential equations with some other machine learning algorithms, trying to predict the viability in combination of different chemotherapies. And here we have, for example, the predictions of the model, and then to validate using cell culture models, basically that. So the idea here is to show a little bit about the specific analysis, not exactly the biological results, but the different approaches that we are working in different models, including imaging, NGS, machine learning, basically that. And of course, this is possible with the work of many people that I'm going to mention all of them, but are part of their giving data, recommendations, the biological context. So I guess I'm very promiscuous about the biological topics. But I am really focused on the bioinformatics analysis.
Bioinformatics and system biology, and he's very supportive. That's why everyone is looking for him. I think at some point he has to hide. Okay.
Thank you so much.
Perhaps Plus, you can tell what you have for
Yeah, I didn't
cancer
bring a
care.
formal presentation, but I just mentioned it with the program. So,
The one
Vanessa
that-
was there in the beginning, is the coordinator. It's okay if you use my phone. Coordinates the program of, it's called epidemiology of cancer, but really we don't do that much epidemiology, we do other types of research. The program is this year 25 years old as a formal unit, let's say, but the research has been going on longer. Now the problem, as you all know, that in Costa Rica, gastric cancer is a big problem. It's going down, like in many other countries, but it's still a big problem. And the reasons are not well understood. This we have in common with several countries in Latin America, especially on the Pacific coast, where the Atlantic coast looks quite different. So the reasons for these differences are quite enigmatic. And that is, well, the gastric cancer here and the reasons for it is of course a public health problem, but it's also a scientific problem, right? So we have two basic lines of research. It's a little bit, very much discussion on the university how you define a line of research, but we choose to make two broad lines of research. One is the etiology of gastric cancer, and the other would be markers of gastric cancer. These are of course overlapping. But very briefly then, what we're doing in etiology, we look a lot at Helicobacter pylori. We try to see, well, the prevalence of infection in different zones and I should mention too that Costa Rica has high and low risk zones, just like you referred to the work of Dr. Correa in Colombia. They have very well defined high and low risk zones. So we look at infection of Helicobacter in these zones, in different populations. We look at the virulence factors that they have, and we also look at the geographic, the phylogeographic origin of these Helicobacter, because we know very well that the East Asian strains are much more virulent and much more associated to cancer than, for example, European or African. I can say that most of the ones we find here, as expected, are of European origin. We do find some African strains. We have found none from East Asia. So we look at that, and then we look at, we have had problems, I could mention that later, with collecting patient material. That has to do with legislative changes in the country. So we have entered into animal models and in vitro models. I'm not going to talk about that. I think Warner and Lucia can explain that better to us. But very briefly, the animal models, yeah, we are already discussing them. And in the in vitro models, it's the work of Lucia, which has to do with, for example, PD-1 expression in response to Helicobacter pylori. And also one of our group members, Wendy Malispin, is doing research on how Helicobacter influences the process of angiogenesis during cell culture, but also in animal models. And the idea is to do it in humans too, but as I said, that has been somewhat difficult. With regard to markers, the markers we mostly study, there are many markers for gastric cancer. Some are good, some are not so good, some combinations are good. Helicobacter is one, obviously, but Helicobacter infection as such is not good as a cancer marker. It can actually be a good prognostic factor. Virulence factors are better, and the phylogeographic origin is mainly of scientific interest. And we also look at pepsinogens, which are precursors of pepsin in the stomach. As the atrophy and of the mucosa progresses, we will find that the expression of different pepsinogens will change. So that's actually like a liquid biopsy to look for atrophy in the stomach. It's not a marker for cancer, but atrophy is associated with cancer, so that we do. And then we do correlations between levels of pepsinogen, cancer, atrophy, etc., and other factors. One thing that we're studying too is polymorphisms, host polymorphisms, and their associations to cancer and to other stomach diseases, basically atrophy and also ulcers. And we have studied a number of these polymorphisms now. We found association with cancer for a variety. Almost all of these polymorphisms are in genes involved in the immune response. We seem to be talking about the immune response. I'm happy because I'm an immunologist. And so we find association with atrophy, gastric cancer, with heat shock proteins, for example, with cytokine polymorphisms. IL-1, Warner was the first who did that in the country and in Central America, in Latin America, I think. And TNF-alpha. We found some associations with toll receptors too, that we're looking more closely into now. And we're associating this with cancer, with atrophy, and with pepsinogen levels. And the latter is because we have a large population of 3,000 persons. Of course, we don't have endoscopy for all these people. It's a general population above 60 years of age. But they come from different zones. They come from high-risk zones and they come from low-risk zones. It's men and women, and they have a lot of other information about these people. The questionnaire is about 50 pages long. So we are associating that to risks for gastric cancer. It has its limitations because we really don't have data on their stomach. We use pepsinogens as a proxy for a real diagnosis, let's say. And that, I think, is more or less what we're doing. What I should mention too is that we have collaborations with, we had with the Finson Institute when they worked more on this, in this line of research, with Dr. Fox and Alex. We work with groups in Porto, in Portugal, especially in angiogenesis. We have several collaborations with the University of Bordeaux and also with University of Paris. I don't remember the number, they have numbers there. And with you, we used to work with Guillermo Perez very much in the University of New York. And due to this problem that we've had and continue having with getting patient material, we've tried to open up, for several reasons really, to other Latin American countries. And we've been successful really in one case only, which is Guatemala. So they used to have a little bit easier time recollecting samples. So we've done projects with them, rather successful, but then in the end they basically ran into the same problem as we do with changes in legislation. But we are working on that. In this July there's a meeting in biannual gastric cancer and Helicobacter meeting in the American continents. This will be in Guatemala, so I hope that we can do some more, develop some new projects then. I think that's, I probably forgot something, but.
It was fine. At least now we have these guys have an ocean. That's the main focus on cancer research program.
Yeah.
Thank you, class. Perhaps one of you, Javier, or maybe you, you talk about immunotherapy as well, tumor microenvironments.
Para que quede la evidencia. Okay. Corrales, sí, Corrales dijo que venía. Okay, well, okay, so very nice to meet you, both of you, and I think I already met all the others. My name is Javier Mora. I belong to the Faculty of Microbiology, and there is a research center that is called CIET within the university, within the Faculty of Microbiology. And also in this center we have like a lab where we have 3 different lines of research, and Rodrigo has one, Steve, who is not here, has the other one, and my kind of group works in tumor immunology, and this is what I will tell you a little bit what we're doing at the moment and which different research project we have currently going on. Then, okay. So first of all, I also work a little bit with tumor microenvironment, especially with the immune part of the tumor microenvironment. And it's already known for all of us that now in the tumor microenvironment, we always get an immunosuppressive microenvironment, which promotes or increases tumor progression and metastasis and also relapse. And now at this moment, there are different strategies. I think you already mentioned some of them, and how to inhibit these kind of things going on here and kind of reactivate the immune system within the tumor microenvironment. And in this context, we are working especially in 2 different areas. One is we are looking different ways to increase immunogenic cell death or to induce immunogenic cell death in the tumor microenvironment through the conventional chemotherapeutic agents, how to modulate this process of immunogenic cell death. And also we are trying to discover, or more than discovery, to characterize novel immune suppressors. And these are like the two different perspectives that we are working and how we are approaching this topic. I have like two main projects working in this part. The first one is related to how to increase the immunogenic cell death. So depending on the kind of chemotherapy that we give, we can induce cell death in the tumor cells, and some of this cell death can go to induce immune suppression, inducing what is called like a tolerogenic cell death, creating resistance to chemotherapy and promoting actually tumor cell growth. We already know that this kind of tumor cell death, this apoptosis-like tumor cell death, increases actually the ability of the tumor cells to grow. And on the other hand, we have also a different kind of cell death, which is called immunogenic cell death, that actually induces immune activation. So our goal in this project is how to modulate or how to create different situations where we can actually induce immunogenic cell death. We're actually working in a project using a drug called metformin, that probably you are heard of it. This is a drug used for diabetes. And actually we are kind of focusing on how the combination of metformin with different chemotherapies like paclitaxel and epirubicin can induce actually an increase in the immunogenic cell death pathway. And with this we have some in vitro models going on at the moment, where we have like co-culture models between tumor cells and dendritic cells, because they are like really important in this process of immunogenic cell death. Actually, the whole process is mainly that the cells are dying, releasing different factors and exposing other factors like calreticulin, and then the dendritic cells recognize these dying cells, and they activate gamma delta T cells and also cytotoxic T cells to kill the rest of the cells that are like resistant to the chemotherapy. So we have also like these co-culture models, and we also like evaluate different markers like calreticulin expression, ATP release also from the cells, to characterize which combinations of chemotherapy with this drug metformin is increasing the immunogenicity of the cell death. We also have some animal models. We have a breast cancer model. These PIME-T cells, this PIME-T is a model of oncogenic modified mice that they spontaneously develop breast cancer. And we have like the cell line that extracted from these mice, and we can do like grafting of this cell line to get a breast cancer model. And we also will in the future establish a glioblastoma mouse model using this cell line here. At the moment we are working in this metformin project just with the breast cancer mouse model to prove like these increases or these changes in the immunogenic cell death pathway. And in the future, we will also get, hopefully, human samples to analyze this. We are currently doing all this ethics approval thing here. And it's a whole story, but yeah, this is for another time. And so hopefully we will get like human samples, and we will analyze also the immune conjecture in the human samples, immunogenic cell death markers, also clinical outcome, and we will also be interested in analyzing also the microbiome of these patients. And these will be patients that are receiving chemotherapy as a treatment together with metformin, because it's already used in some hospitals here in Costa Rica that are already using this drug, because they have seen that the clinical outcome is improved when they combine this chemotherapy with the metformin, and that's why we started to do this project in collaboration with one of the physicians of one hospital here in Costa Rica. Because they have already seen that when they give these treatments together, they have a better outcome, but they didn't know why, or they didn't know what was the mechanism going on, so we started to look a little bit into it. So this is like the first line of the project that we have, and the second one is related to a cytokine that I was working with during my PhD. And this is the IL-38. It's one of the last cytokines described. And during my PhD, we characterized a little bit of the mechanism of the cytokine. Also, we found which was the receptor that was being blocked by this cytokine because this is an anti-inflammatory cytokine. So what we wanted to, or what we want to do is actually to find how this cytokine could be involved in the process of immune regulation within the tumor microenvironment. This cytokine is released by dying cells, and we found that it's released also by dying tumor cells. So what we think is that this level of apoptosis that actually promotes tumor growth is releasing some amounts of IL-38, and this is binding to the receptor, which was also like an orphan receptor of the IL-1 family. And this orphan receptor, we have also shown that is important for immune activation. So what we suggest is that the cytokine is being released by tumor cells and binding to this receptor and inhibiting the activation of an effective immune response. So we want to see also how in patients treated with immunotherapy, how the IL-38 blockade could be a potential new therapeutic approach for activating immune response. So for this, we also have like in vitro models. We will also do this in the animal models. Especially, we are very interested in the glioblastoma mouse model because the receptor that is blocked by the IL-38 is highly expressed in the brain, and also the IL-38 is also highly expressed in the brain. So we wanted to see how this behaves in this model, and also to characterize the potential of an IL-38 blockade in human samples, also getting samples from glioblastoma patients and also breast cancer patients. And we are also currently working in the ethical approval of all this, but actually the glioblastoma project looks a little bit more or closer to start than the breast cancer. But then we will characterize also how is the expression of the receptor, the expression of the IL-38, how this is related with the outcome. So this is basically the two lines that I will be working, or that I am working on at the moment. And we also have still collaborations with a professor in Germany, in Mainz, that has this working a lot with glioblastoma, and also with my former lab in Germany, with Andreas Weigert, he's still working together. We are working together with some people from the Fraunhofer in the IL-38 project, and we still have some collaborations going on there. Thank you very much.
I know that you didn't mention because it's not well cooked, but you have submitted an application for starting working also on lung cancer.
Yeah, exactly. We submit an application, but I just don't like to count the eggs before to have the chicken, or I don't know how to say that in English, but yeah. Yeah, we have a project that actually is like, I think like Francisco is in the project, Ricardo is in the project, Warner is in the project, and myself and some other people, that we have like also a lung cancer project. We hope to get a response soon. This is also with lung cancer and will be like related to analyze in liquid biopsy, like some mutations in the patients, also how they respond to different chemotherapies, and especially associated to the microbiota in these patients, and how the microbiota is changing through the chemotherapy treatment. And we also want to focus this a little bit in this direction of immunogenic cell death induced by this chemotherapeutic treatment as related to the changes in the microbiome that is already described, that some bacterial groups could be really important to modulate this immunogenic cell death response, as well as also another immune responses in tumor patients. Yeah. Mm-hmm.
One of the problems with working with immunological pathways is that they are often redundant.
Yeah.
And so when you do IL-38, do you have any controls for other immunosuppressive
cytokines? We have we don't have it here, but in Germany they have like the knockout mice for IL-38, and we have seen some effects already, like changes in some immune cell populations in the tumor. And what we have checked is like another immunosuppressive cytokines to see if there is like kind of compensatory pathways, but at least with IL-10 and TGF-beta, there is no relationship like changes when IL-38 is gone, that you have like more IL-10 or something like that. But you never know, there is a lot of different possibilities, you know, that could be happening also like to this kind of compensating this system. But yeah, we're still working on it. Yeah, exactly. Yeah. Yeah.
I have lots of questions. I think it sounds very interesting. So, do you know which cells in the tumor microenvironment that's expressing the IL-38 receptor?
Yeah. At the moment, we have checked like in macrophages. There are actually some macrophages, they express the receptor. And actually when IL-38 is binds to this receptor, this reduces expression of IL-6 and another pro-inflammatory cytokines in the macrophages. And also, if these macrophages are blocked with IL-38, these macrophages are also not able to induce Th17 responses in T cells. This was actually one of the papers that for my PhD, and the other one, cells that are, for me, is more important is like the gamma delta T cells. They express, it's really interesting because when they are inactivated, they don't express the receptor, but as soon as the T cell receptor is engaged, they start expressing the receptor for IL-38, and this receptor is essential for IL-17 and interferon gamma production from the gamma delta T cells. And as in the moment when IL-38 is blocking the receptor, you get like a dramatically reduced production of IL-17 and also interferon gamma from the T cells. And this is also what we are working a little bit more with Andreas Weigert in Frankfurt. Yeah. Mm-hmm.
Did you have the IL-38 knockout mice, or you just have worked with them?
We have it. Well, they have it.
So have they been combined with like checkpoint inhibitors and tumor studies to see if it...
I think they will start doing that, but not yet. Yeah. Yeah. Mm-hmm.
With the metformin thing, the effect there, is that related to the mTOR signaling pathway? Because that's one of it's an inhibitor.
Yeah, actually what we have seen and what we think is that the whole effect that the metformin has is related to the induction of autophagy, which is also related to the mTOR. It's like AMP kinase dependent effect, and is related to, because metformin increases autophagy, and also an increase of autophagy is related to increased expression of chireticulin and release also from some pro-inflammatory molecules from the dying cell. So I think it's all related to this increase in autophagy, and this is also something that we will try to demonstrate using this machine that we have here, this image, this flow site, where we can see it's like a cytometry, where we can also take pictures of the cells, and then we can see like increasing in autophagosomes and expression of chireticulin, and this is something that with Rodrigo we are going to establish soon. So, but I think is related in whole this mTOR pathway. Yeah.
Okay. So, but have you tried with rapamycin as well? So
we haven't yet. Yeah, I mean we haven't yet because we are just focusing like in these combinations at the moment, but we have to start trying all this induction and inhibition of autophagy to prove the point. Yeah. Yeah. Do you have? It's okay. Yeah. Okay.
Yes. Rodrigo, do you want to present your fantastic
maybe while you celebrate, I can ask you
Sorry?
one question.
Yeah, sure.
I was just thinking, because with the use of chemotherapy for induction of immunogenic, I know there's been some examples where it seems to work. Sometimes when I think about it, I'm a little bit confused how it will work in a patient, because all the hematopoietic cells, they're also very sensitive to most of these chemotherapies.
Yeah.
You need T cells to really work well, they often need to proliferate.
Yeah.
So I've actually been a little bit
Skeptic.
skeptical about it, if that would really work, but it seems like it's working. So do you have any ideas to why it can still work with patients?
Yeah, I think at the moment what it's being known or is known at the moment is that some treatments like anthracyclines, like epirubicin or doxorubicin, they do induce immunogenic cell death. And also the gold standard in mice is different, because you have like a vaccination with the dying cells, and then you graft the tumor, and if the tumor grows lower or doesn't grow at all, then you have this induction of immunogenic cell death. In patients, it's because, for example, the concentration of the drug that you give is not really killing like all the hematopoietic or immune cells, so it's still inducing like immunogenic cell death in some of the remaining immune cells. But actually, the idea of this project is to combine with other treatments like metformin, that is not a cytotoxic treatment, to reduce the amount of the chemotherapeutical agent, to reduce it to a point where probably it doesn't kill a lot of tumor cells, but also doesn't kill like all the immune cells or the microenvironment, but it still induces immunogenic cell death enough to reactivate all the immune system. So actually the idea of this is like finding a way to combine it with other drugs that promote the immunogenic cell death pathway without damaging all the immune cells. But you have a fair point, because if you have like higher concentrations of the drug to kill like really a significant portion of the tumor cells enough to induce immunogenic cell death, you will be also killing a lot of the immune cells. Yeah. So it's a complicated, and I think like in patients or in human samples, there is not really, for example, there is not like a gold standard to measure if you are getting like really immunogenic cell death or not, like you do in the mice, because you can do this immunization and this kind of thing. But in human samples, it's just like you just measure like immune activation, cytokines, immune cell populations, and so on. But there is not really a gold standard to know if it's really inducing all this immunogenic cell death process. So it's like a big limitation there. Yeah. Mm-hmm. Okay, no problem.
¿Se oye? ¿Sí? Ok. Gracias. Okay. Thanks at all. Actually, thank you for coming. Thank you again, Alex, and you. It's a pleasure to have you here. And well, I have problems, so I have two presentations. So I'm sorry, Warner. I have one for the DC lab, so the more general one, and just to show you a bit of the big picture of what we're doing here, what we're going to do, of the equipment, the resources, and all the facilities we will have to collaborate. And then I have something more specific of our research, more basic research topics. So I'm more cell biologist, but I have this convergence, very nice convergence with Warner and Javier on the immunology part, and we actually, with this topic of immunogenic cell death, it's something really fascinating, because we are looking to some of these convergence zones, because I was actually working on autophagy, and now it seems that autophagy is very relevant for immunogenic cell death, and so we're combining good efforts there. But I will just talk a bit about this later. First, talking about this lab of teaching and surgery and research in cancer. So it's a complicated name. We hope to change it in the future so that it doesn't sound like dendritic cell lab. But for now, it's like this because of docencia, which is teaching, because it started actually here at the vice presidency of teaching, but now we are making efforts so that it's something more parallel to the university and actually related to the social part and also research, of course. So the idea of this, well, this is actually part of the National Plan of Prevention and Control of Cancer. And this is a text copied literally from there. So it's to strengthen the generation of scientific knowledge and technologies required for the integral approach of cancer in Costa Rica, with interinstitutional cooperation, transdisciplinary research, international cooperations, and high standards of ethical rigorosity and scientific. So this is something we're actually trying to push in our country, where we have a health security system which is completely separated from academia. And as you've seen examples of Warner and Javier, it's a long effort to get the project approved and then get samples from the hospitals that are otherwise trash anyways. So we're hoping that this DC lab will become a bridge between academia and also the companies, the pharmaceutical companies that could also collaborate with us. Well, there are three sections, as you can see from the building. The section of transfers of knowledge, actually Francisco is the coordinator of this section. The section of specialized diagnostics, Steve Quiroz is the coordinator of this, and he's not now here, but he's in Germany, but I will talk a bit about his work. And the section of medical approach, let's call it like this. And this is Dr. Marcos Zúñiga is the coordinator of this part. So I will talk more about this, since this is something where we're more related or more into that. And this is actually the third building you have seen there. So this is the structure as we planned it in the very beginning. And the idea is that we could use also these resources for the specialized diagnostics, but also for applied research. So the idea is having samples, colorectal samples or gastric cancer samples or anything you would like to collaborate on. We could then run a series of different tests and also contribute to the patient diagnostics. So this is more or less the workflow of this. So we expect to have samples of our liquid cytologies, solid tumors, blood for liquid biopsies. There will be a section for the collection of this. So this is actually the same here, but just as a functional diagram. And here we have lab for cytology, pathology, automated microscopy, state-of-the-art immunohistochemistry, molecular biology, including also genomics, flow cytometry, liquid biopsy, and also the part of the cluster and and the tissue bank. So we have state-of-the-art equipment. It's already packed there in some boxes. So when the construction is finished, we hope to then install all the part for the pathology, and we have already. So which pathologists are going to work on this, and they are really good guys. This Dr. Yavas, who is also a biologist, and then he became medical doctor, and then pathologist, and now he's in the masters of bioinformatics. So it's a very cool guy. Yeah, he wants to do everything in the lab. So, and of course, it's very important for us, the collaboration with the engineers, and here Francisco plays a very important role, because so we're acquiring also automated microscopes for the scanning of the slides to speed up all these processes. And we'll have then these microscopes attached directly to the computer cluster. And of course, there will be very important to have automated image analysis for all these kind of applications, including fish analysis, where we'll like to have some 3D reconstructions and other, we'll start actually with HER2 and fish and also some amplifications. You have seen also the equipment for the IBIS, in vivo imaging system. So we have already the facilities for creating stable cell lines, sorting them. So we have all the plasmids already for this. And we're right now creating 3 or 5, I think Javier said, 5 different cell lines on lung tumors, breast, no, lung, no, sorry, colon, colorectal, lung, sorry, melanoma, and glioblastoma. Okay, so we have 2 glioblastomas, I think. So we have 5 different cell lines that we could then inject into the mice, and then we can give a substrate and monitor tumor growth over time, so without having to sacrifice them. So we just acquired also a very potent flow cytometer with for 30 parameters, so we can develop panels for different kind of applications, including immunology, hematology. Also, we're starting to set up some collaborations with the hematology departments at the hospitals, because they usually have some very rare clones, and they have seen this already in children, where they treat leukemia for a specific type of leukemia cells. And after a couple of months, they see a relapse and they see another clone. Then they go back to the original sample, and then they see that this clone was already present there, but at very low frequency. So it's important then to have more potent panels to be able to identify the subclones that could be also important in the future relapse. So of course, we're also setting up some international collaborations with people called IncelDX. It's a company. And we're also bringing some of their tests to do some characterizations of tumor microenvironment. And this is by flow cytometry. And this is an all-in-one, including PD-L1 status, inflammatory, infiltrating cells, to type them. It is not that detailed, but this is just for the starting of clinical applications. And this is already tests that are already IVD, so we can already start working with the patients and supporting diagnostics for immunotherapy. And also, the idea also is to quantitate aneuploidy as a marker of genomic instability. So we're also setting up a sample, a test for cytological samples for HPV transformation, malignant transformation, where we will assess viral integration. So the virus gets integrated, and then the E6 and E7 genes overexpressed, and we can distinguish women who have only infection with the virus or really have malignant transformation with integration. So that would be something very useful for them to avoid many biopsies and overtreatment. Actually, we also developed tests for detection of bladder cancer in urine, and this is also part of chloration, also by flow cytometry. So we did something called adaptive genetic algorithms to find which were the best parameters or the best features that we needed to classify this and distinguish normal from tumor samples. And with this, we created a malignancy score that allows us to develop a test with 98% sensitivity and 87% specificity. So there, another equipment we're going to have, and this would be something that we were talking already a bit, is an imaging flow cytometer. We have it already running, and that's very cool equipment because we have the good things from both worlds, for microscopy and flow cytometry. We can have many samples analyzed, but then we can have also the information of the colocalization. We can see, for example, if there is translocation of carreticulin, for example, and in thousands of samples of cells at the same time. And looking at autophagy, for example, that we'll talk a bit later. And this is a very powerful tool that will also give us the possibility of having those images to extract many features and do machine learning and collaborate with engineers for getting more insight into this data. So we also acquired the first NextSeq in the region, the NextSeq 550, and with this we are already able to have all the types of sequencing related important for cancer, including, so Ricardo will talk a lot about this, so I'm not going into details, metagenomics, transcriptomics, and all the omics that will be required. Even whole genome sequencing we can have for the first time in Costa Rica, because there are also other 7 small sequencers, the MySeqs, but those were not really fully equipped for this kind of analysis. So we also acquired this equipment. This is the Rhapsody for single cell sequencing. You still have it still packed there, and actually it's very expensive. So we have seen that it's crazy to have these experiments. So we have to be very sure which kind of experiments we're going to run with this. And but we have it there already, and we're really willing to use it. The liquid biopsy part is also very important for us. We acquired the digital droplet PCR system so that we can assess all these things, but I'm not going to talk in details because Ricardo will. So from the liquid biopsy, we're interested in circulating tumor DNA and also in circulating tumor cells. For this part of circulating tumor cells, we also acquired this Genesis system. And this Genesis system is a microfluidic system where you can detect the epithelial cells. They get trapped into these arrays. And then you can do some stains and then characterize them and then count the cells that are circulating. And then even you can also extract from there and to do some sequencing and then some small panels to characterize this, for example, for prostate cancer or other types of cancer that are very, very related or accompanied by circulating tumor cells. And of course, this biocomputational part, so we acquired the CERCE cluster that Francisco will talk. CERCE was a god-called hurricane, yeah, because it's very, very good cluster. Yeah. And the other one was called earthquake or thunder. Okay, we have thunder, we have hurricane, and see what comes in the future. Right? And well, this is just related to the general part of the DC lab. Now I'm going to talk a bit more on our research topics. So we We actually started with these 3 guys out here, so at Lab of Tumor Chemosensitivity from the Faculty of Microbiology. And so we're trying to combine experimental approaches with systems biology, so mathematical modeling of these kind of things. Actually, Jose was one of our students starting with this on the Masters of Bioinformatics. Our aim, and actually we're taking this aim directly to DC Lab and trying to expand it with all our collaborators, is to generate scientific knowledge to foster personalized therapy by different kind of approaches. We started in the past with testing some chemosensitivity assays, but in the end, well, we realized that it was not really supported by the oncologists, and they were thinking, no, you cannot mimic the situations in vitro, and they're completely right about it. Although there was some evidence that some of these assays could at least predict resistance, not sensitivity, but then it was for us a starting point to work on these topics, and then we realized that other markers could be even more important than cell death, and especially those related to immunogenic cell death. And then the other part on genomics and biocomputation. So just to introduce myself, so I did my PhD in the DKFZ, German Cancer Research Center, and my postdoc in bioinformatics and cytobiology biology at BioQuant, also at Heidelberg University. My research topics directly, so they are involved in molecular sensors for cancer. Here we work in sphingolipid metabolism and also autophagy. And this is actually, I did my postdoc working a lot on autophagy. And now, since I met this immunologist, including my brother Javier, so we're working on this autophagy and the role of autophagy in immunogenic cell death. And also some other sensors that we'll describe very shortly. And then the part of genomics and biocomputation that I will also talk a bit in the end. So this is just an example of, I'm not showing all the data, but it's a very interesting findings here we have on the role of autophagy on immunogenic cell death. And so we have, for this, we have used a sensor to measure autophagic flux. And autophagic flux is a measure of dynamical measure of autophagy. And that's important to mention that when you say that, for example, metformin induces autophagy, it's not autophagy. So you have to characterize the phenotype of autophagy. And here we have, for example, a way to measure autophagic flux. We have a tandem version of this. So we have now better sensors for this, but I will explain you this. This is something with mCherry and GFP. And what happens is actually when the autophagosome gets formed, then it will bind to a lysosome. Then the GFP is pH sensitive. This pH sensitive makes that the green fluorescence is lost, and then you have only the mCherry fluorescence. So with the relative amounts of fluorescence, you can measure this autophagic flux. So we're trying to correlate this autophagic flux with the exposure of some immunogenic cell death markers in this collaboration with Javier, including chlorreticulating exposure and ATP secretion. One on HMEB that it didn't work actually. The kit they told me yesterday was the worst part. But we were working this at the beginning with breast cancer cell lines. So we're interested to repeat that anything or whatever other models you're interested in. And here we have found that with hydroxychloroquine, we have something very interesting. And it seems that hydroxychloroquine is favoring accumulation of autophagosomes. So, sorry, here, autophagosomes. So the autophagosome gets formed and fills the cells, but it does not mature into autophagolysosome. And it might be, so we are trying actually to prove that now, that the accumulation of autophagosomes like this is what renders the type of cell death as immunogenic. So we are trying to suggest that the accumulation of autophagosomes, it's a kind of surrogate marker of immunogenic cell death, at least compared with the markers of immunogenic cell death that Javier is working on with chirotectin and ATP secretion. So we are going to use, actually we're planning to do this experiment in the following weeks. Here we have a sample condition where you have epirubicin plus hydroxychloroquine, and we see that increases a lot of these autophagosomes that are being actually filling the cells, and we would like to see if there's a correlation between this and the cardiac exposure during cell death. Well, other type of sensors, we have created also a sensor for protease activity, and this is something we just published in JBC for dengue and Zika reporter, but actually it's useful for any other flavivirus. This kind of sensor, so the principle applies for any type of proteolytic activity. So if you have any other interest with a protease, we could create a kind of sensor like this. So this works in a way that we have a GFP or an M-Neptune. We have also a Neptune version of this. And we have a linker binding this fluorescent protein to a quenching peptide. This quenching peptide would interfere with the folding of the GFP. But then this linker will be the target for a protease. And the protease, in this case, the viral protease will come here, and then we cleave this linker peptide, and then the quenching peptide will go off, and then we'll have the GFP having the opportunity to refold properly and getting fluorescent again. So we could see this at a single cell level, and also Dulbeco plate forming units here. And so this we could follow over time, and this is a big advantage because we can analyze infection cells over time by the proteolytic activity on those cells. But as I mentioned, we can use this approach for any kind of proteolytic activity. Well, I'm not going to talk about sphingolipids because since we have worked already a lot on this, and also Jose discussed a bit on this, the role of sphingolipids in response to therapy. But I will talk more about this part of genomics and biocomputation, and this is part of the work of Steve Quiroz. He has been working, and this is also mentioned by Jose before, on functional genomics using flow cytometry and cell sorting, where we can then pull a library of lentiviral shRNAs, transduce that in cancer cells, inhibit a gene at the time in one cell, and then do some functional analysis, and then look for the enrichment of those RNAs that were, for example, interfering with cell death or proliferation. With this, we have a further, so he is characterizing these very complicated pathways of cell death and proliferation related to genotoxic damage. And we have a list of interest, genes of interest, but now it's complicated because we don't know what to do with these genes, list of interest. So we want to knock down, we want to regulate, but we would like to do something more related to therapeutics. So we are very interested in microRNA therapeutics, how microRNAs are regulating those genes. Actually, we have different projects on this, different applications, one with Warner. Actually, we have to talk a bit about this. Yeah, because I had to buy the anti-MERS very soon. And so we actually work also together with a guy called Christian Marin, who works a lot on one of those microRNAs against pancreatic cancer. And he has worked also on the generation of delivery systems, and it's very soon to start with clinical trials on this. So actually, well, this is the guy here, and his company is called Speraton. They created a mimic microRNA where it's like kind of a principle where you can replace the sequence and then test other microRNAs. But the big problem with this, so he was very lucky actually because he found one that was active in pancreatic cancer. But this part of the target identification, as we were going to work, is complicated. So those are the type of networks that you will see on these microRNAs regulating some of these genes together with transcription factors. And what happens here is that you have transcription factors in arguing microRNAs that will form negative feedback loops, coherent feedback loops, positive feedback loops, and many other types of arrays that are motifs that are not going into details. But it makes that the network information is not enough. So we need mathematical dynamical models to describe the behavior of these networks and be able to understand what is going on. So for that, we created a biocomputational platform. So this works with any list of genes of interest that you have. So you have a list of genes of interest for anything, and you would like to see how the microRNA regulation works on those genes, and we can maybe try to find a microRNA that regulates them all. We can collaborate. So this biocomputational platform will take information on many databases on microRNAs, transcription factors, and then we create automatically these very large network topologies and also mathematical models to do this our analysis. So this is just an example. I will go very short on, very quick on this. So we identified some genes that are the interesting behavior of dosage compensation, where we have high variation in the copy numbers, but low variation in their expression, either at protein or RNA levels. So taking those genes, we create an interaction matrix, and then we provide mathematical expressions to this to create what is called SBML model. It's the Systems Biology Markup Language. And with this, so we created, well, this is already published. This is part of this biocomputational platform. We started to characterize one of these networks, and this is how the platform will work. This is how the equations will look like. So they are ODEs, so ordinary differential equations. So this is already imported into COPASI, which is the most powerful tool right now in systems biology to work with these kind of large models. And this COPASI model, then it has to be actually fit with experimental data. And experimental data, we initially started using data on the NCI 60 panel. This is a panel of 60 cell lines of these 7 different types of cell lines, including those here. And they are fully characterized, the spectral karyotypes, CGH arrays, gene expression, microRNA expression, proteomics, NGS, long non-coding RNAs, just recently. So we can use this to create many hypotheses in vitro and then go back to the lab because we have the cell lines, actually. We have the actual panel of cell lines here. So we can then go back and then test in the laboratory what's going on. So it's an excellent data set to formulate hypotheses and return to the laboratory. With this data set, then we can train the models. So this takes months at the beginning, and now with the clusters, we hope to speed up all these processes. This is just to show you an example. After many iterations, actually this experiment took like 3 months on a small computer cluster. And so in the end, we get a fitted model with where we can compare the experimental data with the simulated, see the difference, and then do statistics to see if there's any significant difference or not. Here we identify dosage compensation for 2 genes. In this case, it would be MYC and STAT3. And then we can do something called sensitivity analysis to see which model species, which variables are the ones controlling the concentration the most. So having the power or the control, this is actually control theory, and this is a way to know which microRNAs, for example, are regulating the concentrations of those genes. So you would be interested if you see, for example, a list of genes of interest, a microRNA that is being regulated on all of them, for example. You can create objective functions you would like to say, for example, I want to have these 2 genes upregulated, these 2 genes downregulated. You create objective functions, you optimize it, and then you see which microRNAs would be the best fulfilling your criteria. So this is just to show you how we pass from one of these huge networks down to a minimal network of dosage compensation, in this case for MYC and STAT3. And here we found 3 feedback loops. I will just focus on MYC for now. And here we have microRNA 17, 19A, and 20A. There are 3 of them part of the same cluster. And so we determined this doing experiments. These are biologically inspired experiments, so we can go to the model, inhibit each of these microRNAs, see what happens, and do this kind of simulations, and then in the end come to this. Then we took this very small model, we fit it again, and then we saw that it recapitulates actually the behavior of dosage compensation. Then we back to the lab, and this is colon cancer cell lines. So we have chosen 3 of those colon cancer cell lines. And here, because we have one with a copy number of 2, another with a copy number of 5, and this was a copy number of 7. Sorry for the changes here in the slides. And there we could see that if we transfect these 3 anti-microRNAs, so inhibitors of microRNAs, then we'll have this behavior of the cytotoxicity, where we see no difference with the control when we have a copy number of 2. But as you increase the copy number, we start increasing the difference, meaning that probably those cells with a higher copy number of MYC will be more sensitive to the inhibition of those microRNAs. So we have seen also these larger data sets. This is a CCLE. We'll have already a possibility to do this with the CCLE cell lines also. Those are 1,000-something cell lines that we can use for fitting our models also. And we have seen also dosage compensation in this kind of, for these genes in this larger data set. We're currently working on TCGA as well, and to see the relevance of this dosage compensation for patient survival. Also trying to include long non-coding RNAs, another additional layer of regulation because they are molecules able to sponge out the microRNAs. And of course the parallelization of this because the parameter estimation could be really pain in the neck. Okay, and so I want to thank and acknowledge the participation. Well, this is particularly for the work on dosage compensation. It's the work of master student in bioinformatics and system biology, and also the experimental work done by Gloriana in microbiology. But we have many collaborators around the world. For each of these cell lines, we have different collaborators, and this is for the part of the dosage compensation. Professor Pedro Mendes and Stefan Hoops from the COPASI team. Actually, they are the creators of COPASI. And we are collaborating with them for this kind of projects. But as I said, we're very happy to collaborate, and if you have any interest, genes of interest, or proteolytic activity you want to study, or autophagy, or any other related topic, we're very happy to talk about possible collaborations. Yep. Sorry, you have questions that need me to leave the slides here? Otherwise?
Yes. I have a question. Do we go and have a lunch and then we continue?
That's a good idea.
So perhaps we can make a stop then and perhaps, let's say, 1:10 or something like that. Is it okay?
Sure.
Hi, everybody. Well, good afternoon. My name is Ricardo Chinchilla. I'm going to tell you about my two projects that I'm currently starting in the clinical trials. My current focus is in liquid biopsy and NGS data analysis. So the intent of the presentation is to get knowledge about the little thing we are here in Costa Rica. And I hope you have that chance of collaborate with us. One of the clinical trials that we have currently is the genetic markers of circulating tumor DNA for monitoring breast cancer patients in Costa Rica. This clinical trial is to try to get like 3-4 samples in breast cancer in cancer patients that have metastasis in breast cancer, and we get the ctDNA of the blood. Then we try to quantify the concentration of the ctDNA, and then we usually try to get more biomarker of the ctDNA, like the fragment size of the ctDNA. And also we are going to sequence in this gene panel like 28 genes related with the cancer biomarkers like KRAS, PIK3CA, RET, mTOR, TP53. And we try to make to know how is the development of the disease in this breast cancer patient, and try to get knowledge of when emerging new mutation points
in this patient, and how is the time between the first sample and the last sample, how is the behavior of the different mutation in these patients. And the second research is about mutation in the gene MTHFR in the C677T and the A1098C. These polymorphisms are related with the effectiveness of the fluoropyrimidine treatment in metastatic colon cancer. So the main issue with this clinical trial is try to get how is the toxicity of this treatment in the metastatic colon cancer, and how this polymorphism try to get like new biomarkers of how the patient responds to the treatment and how to prevent toxicity in these patients. So these two clinical trials is an initiative of the University of Costa Rica to try to get more clinical trials in the country, and one of the main purposes of the DC lab is to get more clinical trials in Costa Rica and try to get more knowledge and try to understand first how the biomission, and then try to discover new therapeutics with the monitoring of these biomarkers. So it's a short presentation, and I don't know if you have a question for this clinical trial.
How many samples have you collected so far, and what have you done with the breast cancer?
In this one, we have 25 patients, and of these 25 patients, we have 3 samples, and we try to get the 4th sample of this patient to get like a monitoring of 1 year, because we have like 3 or 4 months between samples, or try to get in the research way how the fragment analyzer, the fragment size of the ctDNAs correlate with the metastatic sites of each patient. So this is the main issue.
And there are some patients that just pass away, so you cannot collect all the samples that you wanted to collect, right?
We see that in patients that we only have 1 or 2 samples, the concentration of the ctDNAs is higher than other patients that we can recollect like 3 or more or 4 samples. We have too many analyses to do, but it's a preliminary data, but we see that patients that have more than 2 nanograms per microliter of ctDNA have a worse scenario than patients that have less than 2 nanograms per microliter. But I know that the size of the project is small, but it's like a view of how is the behavior of these patients, and then try to make another clinical trial with more samples.
These are all metastatic patients?
These are metastatic patients, because we try to start with a well-set population that we know the ctDNA are very established and have a good behavior and have a good concentration of ctDNA, and then we try to move backwards and get more earliest stage of the disease, but we try to start with metastatic breast cancer. No, we include all the types, because we try to look first in the metastatic breast cancer, and then we try to segregate in different types of projects, like triple negative, like progesterone and estrogen receptor, or luminal A or B.
Yeah, now it's just a proof of
A proof of concept.
To have an overview of what's going on in order to kind of later on get into more details, right?
Exactly. Exactly, because we try to use the ctDNA for monitoring how the patient is the clinical stage and how to relate the ctDNA concentration with the clinical behavior of the patient.
Sorry, how do you work that with the fragment sizes for the fragmentation according if we metastasis or primary and type of cell death or something with degradation?
It's because different tissues have different endonuclease in the different tissues. So it seems that the different types of endonuclease in the tissue have different size of patterns in the ctDNA. So the main goal is try to correlate the size of the ctDNA with the original or with the origins of the metastatic sites of the breast cancer. We try to get knowledge about if we have like 150 burr pace in the CT, try to correlate with one tissue in the patient that we have clinical evidence of metastatic.
But the difference could be really small, right?
Yeah, this is the problem, that maybe the difference is like 3 or 4 or 5 base pairs in each patient or in each tissue.
And like this with 3 samples over time, can you monitor for the appearance of new things?
Yeah, it's the main purpose to get all the samples, well, NGS, to try to see or discover new SNPs in the second and the third sample that we don't see in the first sample. Is the evidence of new clones in the patient.
So those patients are being treated, right? They're under treatment.
Yeah, they already treatment in the guidelines of the CCSS.
So you could characterize, annotate those SNPs and see if they are related to resistance to that chemotherapy.
Exactly.
All right. So, Diego, perhaps you can jump in. Sure. Thank you, Ricardo.
Thank you.
Yes, probando. Sí, sí. All right. So I did my PhD in biomedical engineering. I came back to Costa Rica like 7-8 months ago. So I'm settling down here and I'm going to show most or pretty much all of what I'm going to show is work that I've done as part of my PhD that's related to using
emerging medical imaging technologies for cancer research applications, both for research and clinical type of applications. The main problem that I tried to tackle was lymph node metastasis detection. And the current situation, or the clinical standard to detect clinical metastasis, is sentinel lymph node biopsy, where you have to detect the location of the sentinel lymph node, which is the first lymph node where the tumor cells will drain. And then you remove the lymph node, take a biopsy, you go to the pathologist and get a result. So there's challenges with this, just the fact that it's an invasive procedure, and the whole, the timeline is longer than the patients would want. As a patient, you want to know, immediately what you have and what you don't have, right? So my goal, or the goal of this work, was to develop or to use a technology that allows you to monitor the status of the lymph nodes and see if they're metastatic or not in pretty much real time. And the name of this technology is called photoacoustic imaging, and it's let's call it like a sister technology of ultrasound. So with ultrasound, you can get morphology of tissue and pretty much like the mechanical characteristics. With photoacoustics, you can study the functional and molecular information in tissue. And just briefly to show you how it works, you use the same system as you would with an ultrasound, and you add a laser, a pulsed laser. So when you irradiate tissue, you are creating like small spots where heat is getting absorbed, or light gets absorbed and then heat gets generated. This heat causes a thermoelastic expansion that is can be read in terms of ultrasound waves. So you irradiate tissue with light, tissue absorbs the light, converts it to heat. Heat causes thermoelastic expansion, and these ultrasound waves are emitted back to the ultrasound transducer. So you can use the same system to emit and detect ultrasound waves, get an ultrasound image, and then emit a laser pulse, get an ultrasound image back, and that's your photoacoustic imaging. So you can overlay both of these images using the same system, and you can do this in real time. The type of information, what this equation means is pretty much the intensity of the image you're getting will depend on how strong your laser is, which is, let's say it's not that relevant at the moment, but then it's going to have information of the optical absorption within the tissue. So literally you can read the color of the tissue non-invasively. And then there's also some factors that depend on the tissue characteristics, water, and I'm going to bring that back in a couple of minutes. So the easiest analogy to represent this is the lightning and thunder. You see the lightning, and then you count how many seconds it takes you to hear the thunder, and then you know if you need to run or not. So this is the same way that photoacoustics work. You emit the laser, and then you start counting from the moment you emitted the laser pulse, and then that will tell you where the stronger signals came from within the tissue. And this is why tissue optical absorption is important, because components or endogenous components of tissue will tell you some information of what's going on. For example, hemoglobin, you can see that oxygenated hemoglobin or deoxygenated hemoglobin have different absorption patterns. So just with these two, you can, for example, monitor tissue oxygenation, which is related in some tumors to the presence of cancer cells. For other applications other than cancer, you can also see the fat content and many other things. But hemoglobin is one of the main characteristics that people have used photoacoustics to study inside the tissue. There's what it's been called the optical window, or near-infrared optical window, which is the section in the wavelengths of the laser you can use that will penetrate best in tissue. And this is because blood and skin and other components do not absorb as strongly. And this is the region where you would want to use contrast agents. So with oxygenation sensing, it's been shown that it relates or it correlates with the presence of tumors. This is not only for primary tumors, but also for metastasis in the lymph nodes. There's studies showing that metastatic lymph nodes also have a decrease in the oxygenation. And let's see. And that's let's call this is functional, functional photoacoustic imaging, meaning that it's showing you function of the tissue, that oxygenation, something's changing, but it's not really showing you molecular information. To see molecular information, that's where contrast agents are the most useful. And with contrast agents, you can design them targeted to a specific molecule or specific type of cell, trying to detect specific enzymes or anything that you want to track and will tell you more information about either the tumor cells or the tumor microenvironment. And so gold nanoparticles are one of the most popular contrast agents for photoacoustics. This is because gold has some good advantages like very strong optical absorption, and it's biocompatible. The chemistry of gold is very easy to stick antibodies and other type of polymers in it. So with this example I'm going to show for lymph node metastasis, we had a small gold nanoparticle, and we used EGFR antibodies that will stick to the nanoparticle, and the goal is that these particles would go and stick to cancer cells that were overexpressed in the EGFR tumors. So we, and the procedure was really similar to the clinical procedure. That's something we wanted to keep because it presumably makes it easier to translate instead of changing the whole paradigm in clinic. So currently doctors will inject a radio tracer in the tumor and they will see how it drains to the sentinel lymph node. With this you would inject another substance in the primary tumor, see how it drains to the lymph node, and then take an image and see if these nanoparticles are accumulating within the cells, showing that there would be cancer cells in that particular lymph node. And the interesting property of these nanoparticles is that if you remember, I mentioned that the near-infrared optical window, which is where you can see the contrast agents better. The nanoparticles in their free form, when they are outside of cells, they will not absorb strongly. This is the red spectrum. They pretty much don't absorb or don't really show up in the images when they're not inside of cells. When they get ingested, then there's an effect where they couple and then pretty much change their optical absorption, so you can see them. So they really are invisible when they are outside of cells and visible when they're inside. There's always going to be some false positives. Maybe particles can stick together. That's where you start using other polymers like polyethylene glycol and some tricks to keep them from, for example, like immune cells taking the particles or sticking to other stuff in tissue. And so then the type of images you would see is when you have particles inside of cells, a particular spectrum will appear. The images, or this particular images, this is a mouse. These two white contours are the lymph nodes of this mouse. This is the neck region. So we were looking at oral cancer with cervical lymph node metastasis. And the mouse was injected with the particles. We waited and scanned over 2 to 4 hours and started to see how the spectrum of the aggregated particles inside of cells started to show up in a region of a lymph node. And we confirmed this with bioluminescence imaging just to make sure that the mouse actually had metastasis. And so this is the type of image that you would build. We are able, if we scan with different colors or different laser wavelengths, we are able to show a map of where there's more oxygenated hemoglobin, deoxygenated hemoglobin, nanoparticles. We could even add 2 or 3 different contrast agents, and if they are different enough in terms of optical absorption, you can split them. And I had a new slide from yesterday. And we can also do some other interesting or useful applications, like if we want to add fluorescence or change the contrast agents, we can add or improve the sensitivity and make it a better sensor. But we can also have nanoparticles that are loaded with a drug. So you could potentially detect where cancer cells are and also deliver the drug using the same laser or using focused ultrasound or other techniques. So you can detect and treat the lesion where you detected it. Also, these nanoparticles, since they absorb light, you can also treat them with heat. So you can use a stronger laser and heat the particles and burn the cancer cells, or ablate the cancer cells in the same procedure. So it will also save a lot of time from detection, biopsy, and coming back to the treatment. So that's pretty much a summary of some of the work I've done and some of the work I plan to continue when I'm here. And that's it. I don't know if there's any questions.
So how, does it matter how deep into the tissue you want to image something?
Yeah, so that's the question that a lot of people ask, and we usually don't want people to ask it. So yeah, there's a limitation in terms of penetration. So it's in the range of centimeters, maybe like 2 to 5 centimeters deep. So if it's something that's very deep, you have the trouble that getting the light in might be difficult. That's like if you really want to see deep, maybe you could do something a little more invasive, like inserting an optical fiber, because ultrasound goes like deep enough. It's the light that's going to affect how good your signal and your image is, depending on how deep you keep going. So yeah, it's in the range of centimeters, but there are tricks that you can play, like inserting a fiber or something like that.
Are there any studies looking to the sensitivity and specificity of these regarding infections or even inflammation when you take a biopsy of a primary? Are there any studies on that?
So you mean sensitivity compared to how the biopsy would do, or?
Yeah, exactly. Like the difference between really this is something that is tumoral or not, or if this is something that could be an inflammation, maybe because of the biopsy that the patient was done, or even an infection, viral infection. Or is there any differences in this sense? I don't know.
Yeah, so there's definitely the contrast agent is the one that would determine how good your sensitivity or specificity is. So if you design it correctly, such that it's very, like the antibodies and how you shield it is very specific to only sticking to cancer cells, that would get it pretty close to what the biopsy would do. Technically, so the way we sell it, we say like it might be even better than biopsy, considering that when you slice the tissue, you might miss like micro metastases. And that's where fluorescence, for example, helps, because then it's brighter and more sensitive. However, when you talk about like functional imaging, like oxygenation, also like with contrast agents, we can see some effects that are caused by cancer. Then yes, like inflammations or even infections in the nodes might be affecting it. So that really is a problem that needs to be kept in mind. But if a good contrast agent is developed and taking all of these in considerations, it might be good enough to overcome those. Mm-hmm.
And the type of tissue, like you showed earlier in the window that there was like the fat had like a lot of signal. So does it affect the reading or does it not? Like
Yeah. So one of the disadvantages of this is that the tissue will always absorb light, right? So any wavelength you use, there will always be some absorption. So when you're trying to detect, hemoglobin, how oxygenated it is, then you run a scan and you kind of estimate it, regardless if there's fat or not. Fat usually has some peaks that are more like farther down to the near infrared. Hemoglobin is more like closer to the UV. But if you use a contrast agent, then you usually want it to be different than the endogenous absorbers in tissue. So if it's weak enough, then it might be getting confused with other things in tissue. But if it's strong enough, like you see this image, this is the skin, and this is the lymph node that was injected or like that got some of the contrast agent. So you can see how bright it is if you use something that's bright enough. Here it's black. If you didn't have this agent, then you could change the contrast of an image, and then you would see signal everywhere, right? So it's not as sensitive as other, like if you talk about like PET, for example, that's like really sensitive. You see signal or you don't, right? Here you don't really see that. Compared to PET, the benefit is like if you had this lymph node in a mouse with PET, you would just see like like a blob of light everywhere, right? Because you don't really see where it's coming. So the resolution here is much better, but the contrast to background is not as good unless you have a agent that is absorbing very strongly.
Do you run any image processing algorithms on those images, or do you do this manually, or how do you interpret and analyze those images?
So we didn't really, or I haven't really gone into image processing that much. So we try to make the images as self-sufficient as possible, let's put it that way. But we do need some image processing, mostly to show the show that the concept's actually working. For example, like sometimes we'll look at lymph nodes and see, this agent is accumulating more in the periphery of the lymph node than the middle, and we want to show that quantitatively. So then we use some techniques to show it and say, like this particular parameter shows that there's a significant difference between healthy and metastatic. The goal, I wish this would be self-sufficient, like I said, like the doctor could just go see this and say like, oh yeah, this definitely is good or bad, right? But that doesn't mean that image processing and all of those like pattern recognition stuff, that probably would still be useful to make it even easier for the doctor to say like, here's the image and here's how likely it is to, be sick or not, right?
We're going to start perhaps carving out, otherwise we'll get out of time.
Which one? How does this photo, this drug delivery system is photo activatable?
Yeah, so one of the, I have like a bonus slide maybe that will be helpful or not. Oops. So there's some agents that we've worked before that use perfluorocarbons. These perfluorocarbons have the property that the boiling points are kind of low. So let's say boiling point like 40 degrees C, right? So within the body, in natural form, they will be liquid, but when you irradiate with a laser, and if there's a photoabsorber that's converting that light into heat, they will heat up just a couple of degrees and then they'll pop. When they pop, you can make it stable enough that they pop and not release, or you could make it unstable enough that they can pop and release some load that they have inside. And the cool trick is that you can release, and also this pop turns liquid into gas, and you can also detect this with ultrasound. So you can do image-guided therapy, tracking whether you really, released your load or not. So yeah, you have to use, you can also make these pop with focused ultrasound, for example. But laser is cooler. Yeah.
And you have all the equipment to do this here in Costa Rica?
Currently, we have half of what we need, and the other half is on its way somewhere in the purchasing department here in school. So yeah, it will be by mid July or something. It should be here.
Will it be here? It will be July.
Yeah. Yeah, so what we have right now is ultrasound, so we can already do ultrasound on mice. For photoacoustics and laser, everything related to laser, we're purchasing the laser right now. So yeah, it will be like maybe 2-3 more months, but it's coming. Yeah.
That's great.
Cool, thank you.
Francisco Siles. Francisco is also an electrical engineer working on cancer. That sounds crazy, but
We're nuts, we know.
¿Qué les va a estar ahí?
Well, thank you very much.
Me robé esto.
Perfect. Well, thank you. My name is Francisco Siles. I'm head of the Pattern Recognition and Intelligent Systems Laboratory. I will talk a little bit about. And I will also head of the section of knowledge transfer in the DC lab. So basically, this is part of what I did here. I did my electrical engineering courses here, bachelor, licenciatura, and then master. Then I start moving around from cables and lights and stuff, and moving into computers and programming. And then I did my PhD in Germany. This is this Doctoral Verum Naturalium in Sciences in Germany. And I started working with football players. So it was quite funny because I went away and people start talking how it's possible that the university sends this guy to study football, But the important thing was the mathematical model that was behind, how you can create algorithms to track objects inside videos or images. And then we start applying those algorithms to follow cells inside microscopy images, for example. And then I finished my postdoc in computational biology. So that means how to apply math, simulations, high-performance computing into biology problems. And then I found these guys that they have this huge problem called cancer. So I'm trying to help the most I can. Well, in the lab, I'm the head. There is another colleague. He wasn't able to come here today, Marvin Cotto. And basically, I'm working with images, videos, 3D objects. I was doing some protein docking in the past in Kansas. And this other guy is working more with speech and text and audio and also data analysis. And this is some of the areas we have been working in the latest years. One of the projects we were working with Steve Quiroz, also one of the guys from LQT, the lab where Javier and Rodrigo are working, is try to classify these images. When you have the possibility to add fluorescence to some cell population, then it's easy to find a higher contrast, to find the background with respect to the cells is easy. But if you're trying to analyze the chemosensitivity, you cannot apply fluorescence because it's genotoxic. Therefore, you have to use bright field microscopy. But of course, there is not enough contrast between the cell and the background. So how can you deal with that? So we basically have been working several years with that. We have just published last year one paper where we reached 75% of classification rate. Compared to the was published, it was 77, so it's 2%, it's not that much, but what was published was with using fluorescence. Our solution is without fluorescence, only bright field. So the idea is to classify that depending on the different phenotypes, apoptosis or programmed death, cell death, and so on. And also we are interested not only in to classify each cell, but also to track each of these cells for the whole video. Because then we can follow each cell and know when the mitosis have occurred and then try to find this cell lineage for the whole video. And then help the microbiologists and the other guys to understand what is going on when you apply some specific drug treatment for the patient or for the cells. There are some other guys working more in image processing, like trying to use radiometric temperature in order to detect differences in temperature in the breast cancer in order to predict what is going to happen. There are some publications in the past that show that this is possible, but there were not enough resolution devices in that time. So nowadays, we're trying to bring that technology back and try to demonstrate that it's possible to detect that. Of course, we are walking, at least, we're not running yet. So we are creating some models where we can control the temperature, we can take these radiometric images and try to develop the algorithm in order to prove that. And going ahead to a possible question, we still don't know how this compares to a biopsy or to the normal treatments, but the idea will be to try to do that in a future work. All that was using images, but also we are working with data, like databases. This is a project from one student we are leading, Warner and me and other colleagues. And the idea is that she's basically taking the TCGA, the database of the Cancer Genome Atlas, and try to find some subtypes of gastric cancer in order to see if there are differences that we can then tackle with different treatments and different classification and clustering techniques. Also, Rodrigo mentioned that we just acquired new equipment, and the idea will be to create like an image facility where we can create these kind of images. We have like a citation where we can add 7 different fluorescence channels and generate this kind of images. We also have this scanner for tissue where we can generate also these images and try to detect PD-L1 and other kinds of tissue. We also have the IVs where we can basically generate this tracking of inflammations or bacteria or cancer cells inside the in vivo mouse. So these are some of the equipment, the citation, the automatic microscope, the scanner, the IVs with the system for anesthesia and all this stuff. And what I said before, this is the project with the Steve Quiroz. The idea will be that this plane here will be the first image where you have all these cells. And the idea will be that the last image will show you the path that each cell has followed. This is the path not only in XY coordinates, but also the phenotype that have suffered the cell during the whole time. There is another colleague, already mentioned before, Jad Abbas, he's pathologist, and the idea will be to use image processing to detect the possible cells that belong to the cancer, those that do not belong to cancer, and inside those that are cancer, then to do the counting automatically, instead of then going for hours counting each cell, 200 cells, 300 cells, and you know you have 3 pathologies, so you have 4 different results, so the idea will be to have objective, you know, less error possible systems. Also, all the other projects that Rodrigo mentioned before, the sphingolipids, so that shows some images, all this, the idea will be to use all this database of images and try to do the automatic quantification in order to help these guys to find these networks that represent this kind of system. So I will be helping in them in the quantification of all these images. Also in this case, to count the circularity of these cells and also the brightness and what else, the density. This was another interesting problem where you need to find this 3D reconstruction for fish and HER2. Normally they have to count the red points and the green points and try to do this difference. But some of these points are probably on top of the cell, not inside. So the idea will be to take some Z-axis cuts with the scanner and try to build this 3D reconstruction in order to get only those proteins that are inside the cells that are of interest. And of course, making the system so they don't need to count anymore. Also with Javier Mora, with the IVIS, the idea will be to have like a library where we can compare the 3D position of whatever we find, because the IVIS can show you some 3D approximation based on some libraries that are stored, but they don't show you exactly the real mouse. So the idea will be to have to be more precise and generate these kind of libraries. And also there is another possible project with Dr. Silvia Molina. She's working with Helicobacter pylori, and the idea will be also to help her count these cells that, you know, I think there are 1,547 cells there. I don't know if you disagree, but of course, this is a lot of work that they have to do. So my work or my part will be to take these images and try to help the guys to make them numbers. And once they're numbers, create models to classify, clustering, machine learning, pattern recognition, and anything that is required. This is basically the contact of the PRIS-Lab, and thank you very much if you have any question. And I can cut you the Wi-Fi if you don't behave. Thank you very much. It was clear or nobody understood, so it's okay. Awesome.
I think now we can proceed with Adrian. Adrian, perhaps he's a young student, just doing his licenciatura thesis with us, and he's going to work mostly in silico with bioinformatics on TCTA, and also with some transcriptomic data that we have from gastric cancer cell lines were co-culture with Helicobacter pylori.
¿Quién se está proyectando? ¿Sí? Ok. Well, hello, my name is Adrián Cabezas Bastos. I'm working with Warner, Lucia, and Jose Molina in a research project that the title is Transcriptomic Characterization of Immune Checkpoints in Gastric Cancer Assisted Biology Approach. I don't have results yet, so I'm gonna talk about the objectives. The main objectives of the research is to characterize through system biology approach the expression of immune checkpoints in gastric cancer in order to identify relevant genes in the immune context. with this, we intend to, well, identify the genes related to immune checkpoints that are overexpressed in gastric cancer, and analyze where that subtype of histologic gastric cancer are more overexpressed these immune checkpoints, and clinical pathological features that this overexpression may have. The first specific objective is to build and to characterize a first-state network of key immune checkpoints using molecular interaction databases such as strings, RepSec, gene expression omnibus. In other words, a first-state network are the molecules, in this case, some regulatory proteins that are directly connected to a set of selected genes, in this case, the immune checkpoints. And the next specific objective is to compare transcriptomic profiles of immune checkpoints in gastric cancer samples from the TCGA database in order to unravel immune checkpoints of clinical pathological relevance. And the next two specific objectives are related with the work of Lucia. And we introduce a factor that is important in gastric carcinogenesis, such as Helicobacter pylori. The third objective is to correlate the expression of immune checkpoints with the status of Helicobacter pylori infection in a dataset obtained from transcriptomic analysis previously performed in an in vitro system, and to compare emerging features regarding the expression of immune checkpoints in the TCGA database and in vitro system of gastric cancer cell lines co-culture Helicobacter pylori. This is the work that Lucia is doing and the work that Silvia Molina also doing. From the results we obtained here, what's the next steps? Well, to understand the role that Helicobacter pylori has in shaping the gastric cancer tumor microenvironment. For example, the early expression that this microorganism may have in gastric cancer, developing new in vitro or in vivo transcriptomic analysis of key immune checkpoints, and study how does this affect patient survival and response to immunotherapy. Thank you very much.
Adrian is just starting. He's
Yes.
just getting familiar with softwares and things like that, and we are giving him specific tasks like curing the databases by hand, and
Yes.
slowly, because it's a small project as compared to these big scale projects of bioinformatics. So he'll be able to kind of cure the data by hand, So he's already working on that. So by the end of this year, he has to deliver his licenciatura thesis. So hopefully it will go well.
There is something. So we have this guy we're talking about, is Guillermo. He's working also with TCGA, and he has already all the pipelines to download all the data and sort the data and have everything prepared. So you can skip quite a big part of your initial work. You can take the data from him directly, because he can give you the scripts to download everything and have everything systemized, because it's a mess. You have seen already. TCGA is a mess, and actually changes from one year to the other. They will change the architecture of the data.
So that's good to know, because I was supposed to know actually, because I'm a part of the committee. But
He
sure,
just
because
finished like 2 months updating the database architecture, because it changed from the last year to this.
well, that's good.
So if you download again the data, it doesn't work anymore.
Then we have to put Adrian in touch with Javier,
Yes.
so he can get the data
I
ready.
can tell him that he can give you all the data on the gastric cancer, so you can already start working on that.
That's good.
Thank you.
And the other thing, will you look for the infiltrating cells, right? This transcript data.
No, not really. Basically, Adrian's work is to kind of select immune checkpoints that have been published in literature. So far, he has searched and identified that there might be around 40 immune checkpoints. Some of them are very kind of not known, but according to the functional analysis, we know that they are immune checkpoints. And try to see how many of those are overexpressed in know, to extract those that are perhaps relevant in gastric cancer. So after the data and looking at in vitro models like Lucia is going to show, we can perhaps start kind of formulating hypotheses to go for the patients, and to try to characterize the expression now in patients, in human patient material.
You just mentioned because there are those algorithms that are able to characterize specific transcript of immune-infiltrating cells, and then you can have like a percentage of cells or the percentage of transcript that is corresponding to the infiltrating cells, and then you can tell if your transcript of interest, the immune checkpoint, is on the tumor cell or on the infiltrating cells.
Okay. Yeah, but I think that's going to be done. We have another master student just starting, Martin Ramirez, on bioinformatics, and he will do this big scale project using these softwares in order to kind of assemble the new landscape based on transcriptome. That's a bigger project. Adrian is a small scale. Yeah. Now Lucia. Lucia is doing her master thesis with us as well. And basically Adrian is performing in silico analysis and Lucia is performing the in vitro analysis on some of those immune checkpoints. And she basically is co-culturing Helicobacter pylori on human gastric cell lines in order to see what's the expression of those when you co-culture those the bacteria with the cells. And now she has
Ya está encendido.
and she has some preliminary data because there are currently some checkpoints that have been regulated when the cells are co-cultured with the bacteria. More or less like at some point Adrian results and Lucia's results will connect. Because hopefully the idea is that Adrian identifies those that are relevant and Lucia will come up to the same checkpoints. That's more or less what we expect.
Okay, so good afternoon. I'm, as Warner said, a master's degree student, and before that I studied microbiology and chemical chemistry, and I made a specialty on clinical immunology. I work at different places at the university, so I'm not going to go much into this because everybody has mentioned everything, and we don't have that much time. So I'm going directly to my thesis project. So my research focuses mainly on oncoimmunology, and more specifically, I'm working on immune checkpoints, just as he already said. These regulatory molecules basically dampen immune responses whenever the receptors on immune cells interact with their ligands on APCs or tumor cells. For gastric cancer, it is well known that the Helicobacter pylori colonization of the gastric mucosa in combination with host genetics and different environmental factors trigger a sustained inflammatory response that results in chronic gastritis. And so in some patients, this gastritis may progress through a stepwise process in carcinoma. And so immune checkpoints on carcinoma gastric cells is described. And what we don't know is how early in the gastrinogenesis they start to express, and whether Helicobacter pylori has something to do with it. And so my question, or our question, is which immune checkpoints are induced in Helicobacter pylori infection early in gastric carcinogenesis? And so with my master's thesis, I'm going to start answering this question with an in vitro model, and hopefully then I will start working on in vivo models. And so the in vitro model consists on exposing gastric cancer cells with Helicobacter pylori at different times of exposures to the bacteria and using non-infected cells as a control. And then what we're going to do is measure RNA expression and measure protein expression. So far, I've just worked with messenger RNA, and I've worked with like 10 different immune checkpoints. And so I'm going to present you the preliminary results of the ones that are most significantly overexpressed. So first of all, I'm sure you all are familiar with PD-L1. And in this model, as you can see, its expression increases over time of exposure to the bacteria, and it reaches a significant point very lately in the infection or post-infection. Then there is this other immune checkpoint, which is very particular, galectin-9, because it is not expressed on the cancer cell membrane, but instead it is secreted. And well, in this model, it had the same behavior or very similar behavior than PD-L1. Its expression increases over time and reaches a significant point at the end or lately in the exposure to the bacteria. The next one, CD24, is actually very interesting because this immune checkpoint has been very recently described, and what makes it so special is that its receptor, Siglec-10, is not expressed on T cells as it's usually described, but instead it is expressed on macrophages. And as we all know, tumor-associated macrophages, or TAMs, are one of the most important immunosuppressive cells in the tumor microenvironment. So we're very excited with this result. And well, its expression behaves a little bit different than the other two. It's not like in a gradient manner, but instead it just starts expressing at the end of the exposure to Helicobacter pylori. And the last one that I'm going to present you is ICOS ligand. ICOS ligand also had a very interesting result, and we are also very excited with this one because, as you can see, its overexpression is much more evident than the three other that I presented, and it is very early in the exposure to the bacteria. So we think these two specifically might be some molecules that we will want to keep on working with, but that's what we have so far. And well, I want to present you the different questions that we would like to answer at some point. First of all, which other immune checkpoints are induced? And Adrian's work is going to help a lot in this question. How does Helicobacter pylori induce this expression? Does it depend on a specific protein? Where does it interact to induce the expression of immune checkpoints? Which signaling pathways mediate this induction? Does this induction change immune cell functions? And of course, the big question we all have is, what does this mean on a clinical level? So as you can see, I have a lot of work to do, but I'm very pleased with these first results because it feels like a very good start. So thank you very much.
So the macrophages coming back to the depression.
Uh-huh.
They were our strength, now they're coming back.
Yeah, Daniel is working a lot on macrophages, right?
Yes.
Yeah, there was this Nature paper last year showing this Siglec-10 result. Immediately we looked for the primers and started to check, because I think all kind of checkpoints are focused on lymphoid cells, and there might be some other cells that could be interesting. With regards to this Siglec-10, it's been shown that if you block the interaction with the ligand based on an antibody, then the macrophages start macrophaging, taking the cells, gastric cancer or the cancer cells up and engulf them and eating them. So that means that probably if you block them, then the macrophages will become anti-cancer and not pro-cancer, as they normally are. That could be actually interesting as well, especially because when you colonize mice, for instance, and there will be a lot of macrophages in the mucosa infiltrating there.
Do you know if gastric cancer has a lot of somatic mutations compared to other cancers? Because I know that's one of the parameters that determine how immunogenic the cancer is and if it's likely to respond. Do you know that?
Well, actually, I think that nowadays they don't use PD-L1 expression in order to give immune checkpoint inhibitors. In gastric cancer, they're using more MSI, microsatellite instability. Yeah, and that's because it is important in gastric cancer.
It's quite important. It's not as big as it is with melanoma or colorectal cancer, but it's like in the middle, you know. It's in the let's say in the top 30 or something like that with regards to detection of melanoma.
Is the presence of H. pylori by itself immunogenic?
That's part of the questions we want to tackle, you know. Gastric cancer everything starts with an inflammation due to the bacteria. So, of course, there is an inflammatory response mounted against the bacteria. And the bacteria has a number of strategies to survive. Even though there is inflammation, the bacteria stays there. So there might be some switching, you know, there might be some tolerogenic response eventually triggered by the bacteria. That's why we want to go early in the process to see, because there has been suggestions coming from a very well-known group, Jerome Gallon group on lung cancer, and they have characterized and they have said that immune checkpoint expression starts like in dysplasia or something like that, a bit earlier than when the cancer is already there, you know. And they have proposed that that might happen in a number of cancers, including those that are more related to inflammation. And this might be one of those cases. That's why we want to go a little bit earlier, because my hypothesis is that probably Helicobacter is changing the microenvironment very early so that the cancer progresses from very early on a immunosuppressive microenvironment. That's what we want to find out more about. Right? Thank you very much. So I will show you a bit of what I've been doing for a long time, and what are the perspectives with regards to the projects that we have going on now, and perhaps more important, perhaps the reason why we came here is try to come up with ideas in order to perform or to start perhaps a collaborative project on colorectal cancer, you know. So actually that was the main idea, try to kind of get to know each other and see what kind of ideas we have that are basic and clinically relevant in order to start with a proposal that perhaps during this year we can submit somewhere in order to start getting money, or yeah, maybe at some point collaborate with the guys from MIT and try to apply for an international grant. I don't know. I mean, it has to start by having ideas and see what happens. ¿Cómo le llaman? Ricardo, ¿se tiene PowerPoint aquí? ¿Qué es Ricardo? Ah, se fue. La de Adrián, vos. Es que como que no me abre el... Uy, ¿qué es eso?
Préstame la suya, Adrián, porque aquí. Usted sí tiene PowerPoint, Adrián. Ahí está la llave. Okay, I will just make a short presentation in order to keep some time to discuss something, you know. Well, I've been working a lot on gastric cancer since I did my master's actually, and you know, I've been doing things from very early in the process of gastric carcinogenesis up to even metastasis. I started actually several years ago when I went to Norway and Denmark, where we actually shared lab with Daniel, studying a little bit on processes of extracellular matrix remodeling during and how does it or how important that is for the process of invasion and metastasis. We know that invasion and metastasis are facilitated by several families of proteases that degrade extracellular matrix. When I was there, I mainly focused on the plasminogen activation system, which is a system that is comprising several proteins. The urokinase plasminogen activator, which is the one that activates by cleaving plasminogen into plasmin, and plasmin has several roles. It itself can degrade several components of the extracellular matrix, but it can also activate several MMPs, which are also degrading extracellular matrix. But in order for this to happen, the urokinase plasminogen activator has to be bound to the membrane receptor called the UPAR. During that time, I basically started focusing and characterizing the expression of UPAR, because if UPAR is not present, nothing of the rest will happen eventually. We started characterizing the expression of this receptor in gastric cancer tissue, and we found that it was highly expressed at the invasive margin, at the invasive front of gastric cancers. But you see also that most of the expression comes from the non-cancer cells. Basically, most of them are macrophages. We also showed at some point on 100 gastric cancer patients cohort that the expression of UPAR on invasive cancer cells, there are a few cancer cells that express UPAR, and those are located basically in the invasive front, just next to macrophages that are also positive. And we showed that if you start counting the UPAR positive cancer cells at the invasive margin, then you start to see how there is a worse survival as compared to those that have no expression of UPAR in the cancer cells that are in the invasive front. At that time, we also came across a very interesting observation, and that was that UPAR, which is supposed to be a protein mainly expressed in invasive tissue, we found that there were epithelial cells in the gastric mucosa that were positive for this receptor, and we tried to see if this could be related to Helicobacter pylori infection, and we found that Helicobacter pylori was very close or even sitting on top of the cells, of the epithelial cells that were positive for UPAR. Therefore, we applied actually for a grant from the Danish Cancer Society in order to further characterize this observation. This was human human material. So we went into a mouse model. The mouse model that normally is used in this case, we used the C57 Black 6 mice that were colonized with a particular H. pylori strain that is mouse adapted, the SS1, the same that guys at MIT use. And you can see how after a while the mice get beautifully colonized with the bacteria. So now we were ready for looking at the question whether Helicobacter pylori in the mice also induced the expression of UPAR. And we clearly could see that in the mice that are not colonized, there is no UPAR expression with except of endothelial cells, which are kind of constitutively expressing this receptor. But when the mice are colonized, then you start seeing the UPAR expression just on the mucus-producing cells that are next to Helicobacter pylori. You see a lot of inflammation also in this tissue, which is pretty much the picture that you get when you colonize mice with Helicobacter pylori. You start to see a gastritis like similar to what you see on a patient. Eventually, we asked what happens if you give anti-Helicobacter pylori therapy, the same as used in patients, omeprazole, metronidazole, and clarithromycin in order to clear the infection. And you see that after 1 week, the inflammation starts decreasing. This is CD3 positive cells infiltrating the mucosa. 5 weeks after treatment, there is no or almost similar to uninfected mice inflammatory response. So it went down. You kind of cleared the Helicobacter pylori. There is almost no infiltration of CD3 positive cells. So they are basically kind of normal. So what would happen then with UPAR? Well, UPAR starts slowly disappearing as well. So there is indeed a connection. So the question was whether it is the inflammatory response driving the expression of UPAR, or this is a direct phenomenon, meaning that Helicobacter induced the expression in a direct manner. That's a bit more problem. We have performed some in vitro studies in order to see, and human gastric cell lines, they do express UPAR when they are co-cultured with Helicobacter pylori. But still, this is work that has not been published yet. We have had problems publishing it because most of the journals want mechanistic studies, and these are more kind of descriptive studies. So we are still trying to get it out. You know that MMPs are also very important part of the figure in invasion and metastasis, and most of the cells expressing MMPs are actually non-cancer cells. And these cells are actually also expressing the inhibitors of MMPs, the named TIMs. There are several TIMs. And some of them actually, you might presumably think that higher expression of TIMs will correlate with better survival, but that's not the case. It's actually the opposite. It's actually the opposite. The guys at PRIS-Lab, where I was, showed that in colorectal cancer, the expression of UPAR, or the TIM1, which is a very important molecule inhibitor, the higher is the expression, the worse is the survival in these patients. This was measured in blood. You can actually run a blood serological test in order to show that. We have also characterized the expression of TIM1. It also, as expected, is in the invasive front of the gastric tumors. Here you see at the messenger RNA level by in situ hybridization, and here at the protein level by just staining. And also we found, similar to other studies, that most of the expression is coming from fibroblast-associated, tumor-associated fibroblasts or myofibroblasts, by characterizing a bit what kind of cell basically are the CAFs, the ones that express this protein. And there has been literature showing that in gastric cancer, if you start counting the intensity of TIM1 staining in stromal cells, that correlates with TMN stage, depth of tumor, and patient survival. And that's exactly what we got in our paper. This is basically, our paper is kind of confirming these kind of results. These are results that we have not been able to follow up because of the problems that we have here in order to get samples. But now that we are apparently almost there in order to get gastric cancer patient samples, I think we will continue giving follow-up on these observations. I have also been involved with some oncologists, Dr. Alan Ramos, for instance, and a master student that we had at some point. She couldn't make it today, into a kind of more clinical record studies. This was a study in which we wanted to see how the chemotherapy does when a patient gets operated by gastric cancer, if those patients get adjuvant therapy, if that improves their survival or not. Basically, what we have concluded is that the most important thing here is that the patient get operated. If the patient gets operated and you get adjuvant therapy, basically, at least in this set of patients, basically what happens is that there is no improved survival. So it means that surgery will do equally well as compared to surgery plus adjuvant therapy, at least in this set of patients. These are advanced gastric cancer, even though, I mean, that's important point to take into account. These were kind of advanced TMN stage cancers. Also, we saw, or we actually wonder what happened in those patients that are unable to be operated. These are patients that did not go to surgery because they were too advanced. And then in this particular case, of course, chemotherapy has an effect. You know, there is an improved survival in these patients, of course, when they give chemotherapy as compared to those that had no surgery and no chemotherapy. That's more or less expected. However, we also saw an important thing, and that it seems, at least in a statistical analysis on this set of patients, that triple therapy does worse than double therapy. And it seems to be that depending on what is the combination between chemotherapies that you are giving, perhaps one is more toxic than the other, and that might be the reason. Because you might expect that triple therapy will do better than a combination of two chemotherapy agents, but that's not the case, at least in this analysis that we performed. We also did a study on, because there has been a question out there all the time, and a number of studies have shown that the ratio between neutrophils and lymphocytes in plasma could actually predict survival. Basically, what is being shown is that the ratio, the higher is this ratio, the worse is the survival. We did this analysis in this set of gastric cancer patients, and we also showed that it is, we confirm basically what literature says. Basically, if you have a higher ratio in plasma samples, that means that you have a lot of neutrophils and not much lymphocytes, then these patients do worse than those that with a ratio less than 5. This is actually interesting because nowadays also this is being evaluated with regards to predicting biomarkers for immunotherapy. It seems also that for immunotherapy, the ratio will predict who responds and who doesn't respond. At least there are some studies showing something like that. Okay, so that's more or less what we have been doing, and this is the project that we are dealing with now. Basically, we call it the ITCG study. Involves a lot of people from different hospitals, involve guys from MIT, and basically is to characterize the effect of Helicobacter pylori infection in the induction and activation of immunological, molecular, and microbial parameters that describe response to immunotherapeutic agents. It has 4 different working platforms. It has bioinformatics, it has in vitro, it has in vivo, or mouse model, and it has human biological material. Where we are now, basically, this took a year, but it's already agreed. We have not signed yet because we had to wait until the Caja Costarricense de Seguro Social gives the final approval, but we are just waiting for that because the lawyers at some point did not agree on this particular agreement. Eventually, it turned out to be okay. We have got recently the approval from the bioethical committee at the Caja Costarricense, and we also have already the approval from the bioethical committee at the University of Costa Rica. What basically is delaying us is that there should be a committee that is created between these two institutions that has to look at the project again and say, okay, yes, go ahead. But basically, all the approvals are basically there. So that's more or less. And also, another project that we are collaborating with MIT is a project that started because we had a problem. And the problem was that at the animal facility, we were colonizing mice with Helicobacter pylori. We had to bring some mice because we realized that the mice that were living in our animal facility were all contaminated with Helicobacter species that were not Helicobacter pylori. And that is an issue because the group from MIT has shown that if sometimes the combination of several Helicobacters with pylori can modulate the immune response to Helicobacter pylori. And we performed some PCR analysis, and basically what we found is that the whole colony of the animal facility of the UCR was colonized with 4 different Helicobacter species. So that's a problem. That meant that we had to buy new mice, put them completely isolated from the rest of the mice that we have, but the problem eventually turned out to be into a project. What happens if you have mice that are co-colonized, pylori plus something, compared to mice that are only with pylori? Will this indeed show a modulation on immune response? That's the project we are doing now. And as part of that, Alex is going to help us on this because basically we want to see the effect of enterohepatic Helicobacter species, and Alex will characterize the microbiome on these mice, basically. This is a project that has been, it's already approved, and the aim is basically this, to analyze fecal samples from the vivarium, from the animal facility, to compare the microbial diversity observed in Costa Rica to the MIT and see. Also, I will be looking at the inflammatory response, specifically looking at the CD3, how infiltrated are the mucosa of these mice that are having 2 Helicobacters as compared to the ones that have only Helicobacter pylori. So that's something that is part of the agreement. We have collected already all the samples to send to Alex, but we are just waiting for the agreement to be signed in order to send them. Ultimately, basically what we do, or we want with MIT, is that we better understand the reproducibility of the animal models for studying Helicobacter pylori related diseases, basically, across institutions, and to identify microbial factors that influence somehow inflammatory processes in the gastric cancer or the gastric carcinogenesis, basically. So that's more or less what I have been doing. Okay? No sé, I don't know if there is any question, otherwise it's fine. Okay. And, okay, sorry? Yeah, well, Alex can tell actually.
I don't think the association is that strong. I don't think I've seen it. They're trying to associate other bacteria, but not with pylori. And pylori doesn't really colonize outside the stomach. So I think that's
other species of Helicobacter.
we try to say that in our mouse models we do. In humans, not the empiric Helicobacter.
Cancer, it's nowadays almost the 3rd in incidence, very important with regards to mortality. And Dr. Corrales can tell, is increasing rapidly in Costa Rica, both incidence. There is every time more and more colorectal cancer here. And this is how it looks more or less with the years. This is a trend, and you can see how it has been from 2000 until 2015. Important increase both in incidence and in mortality for this cancer. Also, it seems like it's not equally distributed across the country. It seems like there are some hotspots for colorectal cancer, mainly Central Valley perhaps, somewhere here. Both incidence and mortality follow more or less the same geographical location. It seems like Cartago has both gastric and colorectal cancer. That's why now Early Detection Gastric Cancer Center, they are also starting with the screening on colorectal cancer. They are interested on studying this a bit more. There are different ways of getting colorectal cancer. There are different pathways, but of course most of the cancers come from adenoma. 85 to 9% are sporadic colorectal cancer, and they are chromosomal instability high as compared to other pathways that are out there that are a bit less common. So there might be different etiological differences. Also, according to anatomical location, it's interesting because the cancer at the proximal colon is fairly different as compared to the distal colon, and so it is with the rectum. They have different biological features, which makes it actually interesting. I don't know, or at least not myself, know what is the most common type in Costa Rica, if there is a more common type or is equally distributed. I don't know. There might be, of course, different risk factors according to the anatomical location. That's questions that I, at least myself, don't know how it behaves in Costa Rica. There are a number of risk factors where obesity is probably one of the most recognized risk factors. We know that our population is getting obese, so that might correlate also with the increase in incidence, perhaps. And of course, the microenvironment is an issue. This is a nice paper because they proposed a model where the tumor microenvironment has to be looked in layers. They say that this looks like a Russian doll, like a matryoshka. And they say that you have to take into account different layers of this microenvironment or even the systemic environment of the patient in order to look at the whole thing. So, you know, if we start with a project, of course, I would say that it's for sure one has to look at the tumor microenvironment and more systemic issues that go around. Also, microbiota is very important. There are a number of papers suggesting that this particular species is important. It is being found enriched in colorectal cancer tissue relative to normal adjacent tissue. It's been proposed to promote colorectal carcinogenesis by contributing to immunosuppressive tumor microenvironment. That's a proposal that is circulating around. In vitro, it's also been shown that it promotes cell proliferation, and in mice, the presence of this particular species in patient-derived xenografts increases tumor growth rates, according to some papers. So basically what they are suggesting is that this species of bacteria actually hits on several parts of the biology of colorectal carcinogenesis. It impacts cell proliferation, it impacts chemoresistance, it impacts evasion or anti-tumor immune evasion. So there might be several ways in which this bacterial species is impacting on the risk of colorectal cancer. It has also been shown that there are some other bacteria that might create biofilms, and this coats the tumor and recruits other bacteria in order to kind of strengthen this biofilm, and these bacteria also recruit immune cells and promote inflammation. So I think there's for sure that one has to include microbiota as part of the equation if you want to put up a project on this colorectal cancer. Alex told this earlier today, that this Escherichia coli pKa is positive is also important because apparently it induces mutations on DNA, particular because this pKa is positive produces these colibactins, and these colibactins can be mutagenic, can induce adducts on DNA, and then the mutational rate will increase. So there are a number of, indications that microbiota plays a role, and in particular there might be some particular species that might be more important actually in the picture. This is a paragraph from this paper. Basically, they say, given the geography, diet and environmental exposure and lifestyle choices all influence the composition of individuals' microbiota, it is important that such data or metadata are collected and studied in global colorectal cancer cohorts. So I guess that a project on colorectal cancer will be actually good if we include dietitians to kind of also take into account that part of the picture, because we know that fiber, and depending on what the people eat, that can modify the microbiota composition. There has also been interesting findings with regards to secondary prevention or even screening, and they, of course, this is the most accepted, the fecal occult blood test, for basically checking for small amounts of blood, which can indicate the presence of precancerous lesions or even cancer. However, this has several kind of variants, and an emerging screening method is actually multi-targeted stool DNA test. It combines the detection of hemoglobin and also detection or isolation of DNA in order to identify mutations that we know that are part of the carcinogenesis process on colorectal cancer. So one can perhaps start isolating DNA in order to find the mutations that are apparently linked to particular stages of the development of colorectal cancer, like these ones. This has been characterized long ago, that we know that loss of APC is an important thing. So if in a fecal sample you isolate DNA, start to characterize mutations, that also can be interesting for a project, to start dealing with the genomics and the mutational burden during the lesions as a way of looking at what's happening during the carcinogenic process, and if there are mutations that are connected that you can use actually to predict if the patient will continue further or is stopping at some point, things like that. Of course, it's much more complex. It's not only about genes, but it's more about genetic pathways. But of course, with new technologies, with sequencing and all these equipments that we have here, it is absolutely possible to screen for a lot of mutations from DNA isolated from fecal samples, for instance. So basically, I had these ideas in order to start up on something perhaps. So it would be nice if you can have your say.
Just a technical thing. Do you want me to record this? Because we need them to
No.
move around the mic, or we can stop it?
We can stop it right now.